Your Majesty, many thanks for visiting the agency. I'm Stefan Tierstrup, I'm the Chief Medical Officer here and the host of today, so to speak, at least the chairman of what is going to happen. We are so pleased having you here. So to start today's conference, I will first give the floor to our Executive Director, EMA Cook. Please, EMA.
Thank you very much, Stefan, and thank you all for being here. It's really an amazing sight to see so many of you gathered here together. So, good morning, everybody, Your Majesty, dear friends and colleagues. Welcome to the European Medicines Agency. I need to find an Irish. Fáilte welcome, bienvenue. It is a pleasure to host you here at our Amsterdam offices. This is a very special occasion for us as we celebrate 30 years of strong science in our 4th office location. We especially welcome our former executive directors, Fernand, who's online, Thomas, Andreas, and Guido. I'm also delighted that many of our former management board chairs, Strawn, Krista, Lorraine, and Rui could be with us in person today, and I know that others are online. We welcome also our chairs of committees, our experts, our healthcare and patient representatives, and many of our staff, both past and present. Thank you all for being here. And let me also give a warm welcome to colleagues and friends who have not been able to join but are joining us online. Today, it's about celebrating science. We celebrate EMA's history, its story, and what it means for public health in the European Union. Your Majesty, thank you for hosting us in your country. We have now been in Amsterdam for 6 years, and it's our new home, and we are very pleased to be here. Because really science runs in the DNA, in the Dutch DNA. This country is home to 13 Nobel laureates. The Netherlands is responsible for the invention of the microscope, the existence of vitamins, and the discovery of bacteria and viruses. And these are all achievements of Dutch scientists. Now, unfortunately, our DG Sante commissioner cannot be with us in person today because of a last-minute conflict, but we are delighted that so many commission colleagues are with us, are able to be with us today. It is really our partnership with the European Commission, now especially DG Sante, that has allowed EMA to fulfill its scientific goals over the last 60 years. Now, today we are celebrating our 30th birthday. We are looking back and we are looking forward. When EMA was founded in 1995, the world was a very different place. We were processing paper-based applications, huge boxes of files, struggling through data and information, trying to make our way, understand what was being presented. In fact, we have an archive exhibition on our ground floor that we hope you'll have the chance to look at later. The filings were quite difficult. They were overwhelming, and we spent thousands of hours trying to make our way through the data and graphs to make sense and see whether the evidence did support the authorization of these medicines. But we felt like pioneers. Our goal was the first ever truly European system for assessing medicines. A body that would serve, promote, and protect public health. It would mobilize the best scientific minds from across Europe to generate the highest quality evaluations to make a difference to millions of patients' lives. We were building a network that would become the envy of the world. Now, before COVID, despite our scientific successes, few people had really heard about medicines regulation. And then everything changed. The pandemic thrust us into the spotlight, and suddenly everyone turned to us for answers. And I am proud of the answers that we gave. I'm proud of the science that we enabled and the vaccines and medicines that we made available to millions of European citizens. In the last 30 years, the medicines EMA has approved have changed people's lives. In the past 5 years alone, we have recommended 430 medicines, including cancer treatments, Alzheimer's, and the first gene editing treatment using CRISPR technology. For animals, we have recommended 40 new vaccines, and in addition, we've done extensive work on antimicrobial resistance. We have broken down barriers to enter a new age of international collaboration where regulators connect, share, and align to improve public and animal health. Now, what difference does that make? Well, last week I sat on a panel with Jimmy. Jimmy is a sickle cell disease sufferer whose life has been changed by CRISPR technology. Literally, he went from intense pain and bedridden to climbing Kilimanjaro. This is the type of difference that we can make as regulators. And every day we make a difference to the life of a patient is a day we should be proud. Science makes our life better. We know more about our world and ourselves because of science. We are better as people. And we must never take it for granted. We must celebrate it as a vital pillar in our democratic societies. And this is up to us, the scientists, to do just that. Because what happens if we don't celebrate science? We don't learn. We don't connect. We don't appreciate the fundamental role it plays in our daily lives and how it helps us progress and serve our patients. So this is what today is all about: celebrating science, sharing our stories, and learning from one another. So please embrace the spirit of learning and discovery today. Let us engage together in this wonderful program. Now, science and medicines regulation is all about continuous learning. And as my daughter says, every day is a school day. So since we came to the Netherlands, my staff have been working on their Dutch skills. And I have been secretly engaging with Duolingo. And Duolingo thinks I'm great. But I'm not so sure. Majesteit, collega's en vrienden. Wij hopen dat jullie genieten van deze viering van de wetenschap. Welcome to our celebration of 30 years of science.
Well, thank you very much, Madam Executive Director, ladies and gentlemen. It's great to be here with you, coming from Fortress The Hague and making it out of there today. That was the biggest gift, I think, of being here today. But I would like to take this opportunity to say a few words and express my support for you and the important work you do. 4.5 years ago, I also visited EMA, not physically, but digitally. I remember it well, and we're in the middle of the corona pandemic. We're also in lockdown. Here in the Netherlands, we've just begun administering new vaccines, which, after thorough assessment, had been authorized by EMA. I was briefed on the full scope of your work, and I thought, what a blessing to have EMA, and how wonderful that this European organization has been based in the Netherlands since 2019. All the knowledge, all the experience, right here on our doorstep. You safeguard an essential common interest, medicine that protects our health, with authority and clarity for 450 million Europeans. I'm extremely proud of the fact that EMA has found its home in the Netherlands. What characterizes our health care is high standards, innovation, and the pooling of expertise. In every one of those aspects, you are an inspiration. And actually, I would like to add to that. I was in Osaka a couple of weeks ago, and we visited the Nakanoshima Cross, regenerative medication. And one of the reasons why they collaborate with the Dutch is not only because of our scientists, but because EMA is here in the Netherlands. You are essential also for our scientists and our industry, so thank you very much for that. So that's why I'd like to congratulate you all with your 30th anniversary, and may I wish you every success for your extremely valuable work which you carry out. And I look forward to seeing you again. And now I have the honor to push the simple button to do something very important to open your conference. Thank you very, very much.
So good morning and welcome everybody. Many thanks, Your Majesty, for the kind words, especially thanks for being here. We will go into the program of today, and as EMA alluded to, it was planned that our Commissioner of Health and Animal Welfare should give some words. He has unfortunately been dragged into something business-like, I guess. Hopefully not as important as this one, but apparently important enough to be dragged away. But we are lucky to have Sandra Gallina, Director General for the Health and Food Safety at DG Santé, to provide the speech that Monsieur Olivér had prepared. So please, Sandra.
Thank you. So, Your Majesty, dearest EMA, I think the Director. So, ladies and gentlemen, it's nice to see this full room. I'm very honored to give you the speech on behalf of the Commission. Today we celebrate 30 years of achievements. I've seen many of the people who contributed to these achievements. Basically, we have, thanks to EMA, I would say safe, effective, and also high-quality medicines. You refer to the vaccines, a big success, but there are many medicines that go untold and they save life. Now, I would like to congratulate you all, the people who still work in EMA, and those who worked in EMA, because, you know, this is an important anniversary because you need to take a bit of stock of what you have done over the years. Both science and our regulations have significantly evolved over the years. This has impacted also on the responsibilities of this agency and much broadly. What has remained consistent is the good relationship that this agency has with the Commission. We are in the first year of the mandate of this Commission, so many plans, ambitious plans are there, and we're counting on EMA to help us. There is a vision for a European Health Union. The word union may evoke some feelings. The European Health Union should protect citizens, should be about giving citizens the possibility of being protected from health threats. Now, we also need to ensure that citizens have access to affordable medicines, and here the key word is affordable. So we have the quality, we have all the elements, but we need to have also the possibility to access the innovative part of the spectrum at affordable prices. Now, this requires a competitive, robust pharmaceutical structure. So we have to have, both for the pharmaceuticals and the medical device sector, and biotechnology, let me not forget that, a competitive industry. Since 1995, when EMA came to life, we have had, as EMA has been alluding to, the bedrock of science here, which was good for us in the Commission. We have worked together also to supervise the use of medicines. But, you know, I would like to say that EMA today is much more than a regulatory body. EMA today is trying to foster cooperation, is trying to ensure that our healthcare systems can benefit from innovation, but the EMA is also looking beyond the pond, beyond Europe, and this is very, very important for us. Of course, EMA's main contribution remains ensuring that medicines in the EU are safe, effective and high quality. The sound and robust assessments by the agency and scientific committees are the basis, I repeat, for the Commission decisions to authorize the medicine. So it's vital that these assessments continue to be done in the highest of standards. I would like to say that in 2024 you beat all records because you authorized 100 new medicines, which is the first, is a real first in all the years of, I would say, life of this agency. Now, I think that together with the member states, the Commission, the EMA, is trying also to give life to a part that is vital. It's the clinical trials. And you are running the clinical trials, I would say, information system, the CTIS. This is quite an important element because trials is also about patients and how they can participate in innovative, I would say, tests, but also life-giving possibilities. Now, I would like to say that EMA also tries to give us a hint of the state-of-the-art technologies, try to identify the risks, but also the potentials there. And I would like to stress that the oversight responsibilities of EMA are key in this respect, because, you know, many scientists try many things, but it's important to get then an assessment, a real assessment. So we're very grateful for that. And the assessment goes for the whole life cycle of the medicines. It's not just one moment. Now, I want to refer here in particular to the inspections that EMA does for the manufacturing sites, which ensures that all companies in Europe and beyond, do live up to the very strict good manufacturing practices of the European Union. This is very important because it means that there is a bit of a uniformity in terms of production. We are all talking about competitiveness, but you know, competitiveness starts from having the same rules. Now, EMA got a new mandate, Your Majesty, under 2022. And it means that EMA is now doing a bit more than it was doing in the past. And I want to refer specifically to a task that's very dear to me, is the preparedness. You spoke about COVID. We need to be ready for any emergency. CBRN, your NATO summit, is talking about things that we are also talking in DG Santa. We have quite a strict relationship with the NATO authorities in terms of health, unfortunately, I would almost say, but it's the reality. And here I need to say that we have a good role for EMA in managing shortages, which is a key word of some of the problems for patients. EMA is also very active on antimicrobial resistance. I will not go into the whole of the details, but this implies that they are active also for not just human medicines, but also veterinary medicines. This is the whole concept of One Health, you know, that we are all healthy if we start stopping, I would say, zoonoses that may crop onto us, but also not overusing antibiotics with the animals. Now, a small word on medical devices, because I think it's quite important that I refer to that. We have medical expert panels here in EMA. It's very important because there are some medical devices that are high risk and they are life-saving, and it's quite important that EMA also got this expansion in its role. It didn't have this many years ago, so I'm looking forward to the next years. Now, final word needs to be for the initiative that my commissioner launched within the 100 days, as promised, is the Critical Medicines Act. Now, shortages require, I would say, urgent action to have, I would say, production of all the products that we need here in Europe. We hope that the Critical Medicines Act, together with the pharma reform, will be a success because the two parts are somehow complementary. But EMA is already doing a lot because they are running the European Shortages Monitoring Platform, which will become, we hope, will be solidified with the Critical Medicines Act. This is still in the minds of the co-legislators, I suspect. And in any case, EMA is always developing guidelines for the shortages, doing, I would say, some also very good initiatives to try to fill the gaps where they are. All of this is voluntary and once again comes out of the solidarity that member states are showing. And I see here many member states represented that were very strong in terms of solidarity during vaccines. A word on biotech. The Draghi report, you may have read it, was saying that, there is a lot of work on advanced therapy, medicinal products in Europe, and that there is a great potential for breakthrough technologies in Europe, but that we are losing ground to the US and China. It's a fact. It's a fact. You look at the numbers. So I would like EMA to be more and more involved into the biotech side. We will possibly be working together there. And we as Commission, we will be launching a very ambitious Biotech Act to make the most of the biotech revolution here in Europe. So looking forward to that work too. Needless to say that biotech holds a huge promise for all of us. It's a fantastic field, but it requires quite a lot of investment, as we all know. So it remains me to say that I thank you all. I congratulate you for all your success. And I would like to say many happy returns to, I would say, EMA, and thank you for listening to me in this humble shape. But, I'm glad that I didn't have a cough, because I'm terribly cold, and my dread was that I would collapse in front of you, Your Majesty. So I'm very glad I made it. Happy returns to EMA.
Many thanks, Sandra, and thanks for not ending up in a big cough here. That would have been that nice. So moving on in the program, we come to our keynote speaker. And in planning today, I wanted to find a keynote speaker who had made a discovery in basic science that had led to development of medicines for the benefit of patients. And I'm pretty sure you can all think about different topics, Nobel laureates and so forth. But I'm pretty sure all of you have heard about GLP-1 antagonists, or maybe heard about them under their trade names like Wegovy and Ozempic, all coming down to discovery of GLP-1, a hormone of the pancreas. And therefore, I'm delighted to give the floor to my fellow Dane, Juul Holst from the University of Copenhagen. Jens is a professor in human physiology and among the discoverer of GLP-1 and its meaning in human health. It's really a pleasure to have you here, Jens. You are a renowned scientist. You're also an example of people working a bit below normal retirement age. But we don't speak more about that. Please have the floor, Jens, and here's the clicker for you to move the slides forward.
Thank you very much, Stefan, Your Majesty, ladies and gentlemen, colleagues. I'm very pleased to be here today, and after what I've heard already, what I'm going to tell you, I think fits nicely into the topic of today. So let's move on to it. It's basic science to new therapies, the discovery of GLP-1 and its role in human physiology. So the background is that, we started this work back in the 1970s, and that time obesity was not a problem in Europe at all. And there was also diabetes, but it was something with old people's disease, and there was not a lot you could do about it anyway. No, peptic ulcer disease, that was something that was important. I came from surgery, and we were very interested in this. And some of our patients, it turned out, after the operations, developed hypoglycemia, too low blood glucose. So it was also clear that this was because of insulin. So this was my job to try to find out why was that. Here you see an example of it. You can see in the top, the glucose dives very deep down in hypoglycemia. And this was because of the insulin release. So something that we did with the operations caused this. And it was clearly coming from the gut because it was only in relationship to meal intake that this happened. So that was the job we started out looking for. And I was immediately attracted by these cells. These are cells in the human gut. They're lighting up with an antibody against glucagon. Now, glucagon comes from the pancreas, of course, and not from the gut. So this is strange. I mean, why was that? Why on earth would there be glucagon in the gut? But on the other hand, glucagon was known to create insulin secretion, so it could be a candidate for this. So we put up this hypothesis, and the reason I show it is that it's from 1973, that something from the gut was responsible for this reactive hypoglycemia. On the graph, you can see the hypoglycemia on the top, and then you can see the stuff that we were measuring. We could measure it already then, that is increased in secretion after a meal intake in these individuals. So we said that this is probably the reason. And it took 20 years, and it was true, but I mean, that's another story. But we found out what was in those cells, and that's the interesting part. It was a peptide. It's called glycentin, 69 amino acids. In the middle, it has glucagon, and that explained why it was the antibodies was reacting with this peptide. But it didn't do anything in itself, so it was not the solution to the solution to the question. On the other hand, it was interesting because it seemed to be a precursor for glucagon. All peptides are produced via precursors, and we could find the bits and pieces also in the pancreas. So this was very likely to be the precursor for glucagon, and people were very interested in precursors these days. It's very exciting. They could cause the release of even more than one hormone. So this is really, really interesting. And then it turned out that proglucagon probably was a little bit bigger than our 69 amino acid peptide. Something was missing, was probably twice as large. So what was in the rest? That was the interesting part. And then there was an American molecular biologist. This was the day when molecular biology all of a sudden made its introduction, and he cloned the cDNA, the coding nucleotides from hamsters. And you can see the structure up there in the middle. To the left, you have glycentin. This was correct. Glycentin was a part of the precursor. But then there was also something else there. There was a stretch that was glucagon-like 1, and there was also an additional one, 2, GLP-2. So this was exactly the information we needed. So we immediately synthesized those stretches as peptides, made radioimmunoassays, antibodies, and started looking for these peptides. And a long story short, what we came up with was that this precursor, which is shown on the top, is processed, as we call it, cleaved differentially, depending on whether this happens in the gut or in the pancreas. In the pancreas, all of these stretches with the glucagon-like peptide 1 and 2 are together in one big molecule that doesn't do anything. So that could have stopped the story there. That would have been the end. But on the other hand, in the gut, they were cleaved out separately. We could show that very clearly, and we could also show that they were secreted. And now we could test them to see, are they doing anything, these peptides? So we did that, and here is my favorite tool. This is a pancreas from a pig. It is very good at making pancreatic juice, as you can see, but it also produces insulin, of course. So we could test it, and we tested GLP-1 and GLP-2, and they did absolutely nothing. So again, but what we did then was to say, okay, let's go for the natural peptide. Let's take some extracts of intestines from pigs and from people and use our assays and try to look for it and sequence and do all sorts of chromatographies and purify and so forth. And we ended up being able, we got a pure peptide, we could sequence it, and we could test its effect. And now we had insulin release, very, very powerful insulin releasing peptide. And that was, of course, wonderful. So that was the immediate discovery, of course. We had this insulin-releasing peptide from the gut, but there were many things that could increase insulin secretion. But this one was special because it also inhibited glucagon secretion, and as you can see here. And this is important because people with diabetes, they have too much glucagon. That's one of their problems. But this one could inhibit it. And that means that if you have a problem with a high glucose, then you'd like to have insulin doing the job. Insulin is released from the pancreas, makes the blood glucose go down. If you have too low glucose, you have glucagon that will keep up the glucose by increasing the hepatic glucose production. So by having an effect on both glucagon and insulin, you would have a double effect on glucose regulation. And that's exactly what we had. So we looked at this, we infused people with GLP-1, as you can see here in the bottom, and then looked at glucagon concentrations, and you can see that they were inhibited. We could look at insulin, it stimulated, and as a consequence of that, in the middle, we could look at the glucose production in the liver. And it fell by 30%, and that, of course, led to a decrease in blood glucose, but not very much, only by 1 millimole at the most, because these effects were both glucose dependent. You couldn't create hypoglycemia, not very easily at least. So this was new and interesting. So we had a powder that had tremendous effects on glucose. This was interesting, of course. What else did it do? And we spent a lot of time trying to find out what this white powder might do in people. So here's one of the crucial experiments we were looking at: appetite regulation. And as you can see on the top, you have an increased satiety between meals if you infuse people with it. You have a decreased hunger. And as a consequence of that, you had decreased food intake after you had infused them with physiological levels of this hormone. So this was apparently one of the physiological actions of it. And here's an interesting thing. It was dose dependent. The more you could infuse of the peptide, the more people would stop eating, the less nutrients they would take in. This is important as we move ahead. So how does it do that? Isn't that interesting? Is it on the stomach? People say that some of them vomit afterwards. No, it's not on the stomach. Could it be something with the vagal afferents? That's very likely. We won't have time to discuss that. But there could also be central effects. And we looked at that by injecting rats with radioactive GLP-1 and simply looked for it. Where did it go? And it went to this region of the brain, which is called area postrema. This is what is called one of the circumventricular organs. That's sites in the brain where there's some communication with the periphery. And that's where it went, as you can see here. And also, there were receptors for GLP-1 in exactly the same tissue. And even more importantly, if you look at activations of the brain neurons in this region, you can see to the right that they stain with something called C-FOS that indicates activation. And when you give them it, you have the activation. So this is how it does. And there are regions of the brain that are then activated. For instance, this region, which is called amygdala, I'll come back to that, and hypothalamus, where we have the traditional centers for regulation of food intake. So this is what it looks like. These are slides I borrowed from the Novo Nordisk company. To the left, you can see where does semaglutide, the famous one, go. It goes exactly to those circumventricular organs I said. And to the right, you can see, let's see if this works, it does. And to the right, you can see which neurons are activated. And again, you have hypothalamus and amygdala, again, and the nucleus of the tractus of the solitary tract. And these are the regions that are activated. And that's very important, because what are we now talking about? We're talking about the reward mechanism, the reward mechanisms. And here you have amygdala and accumbens, which are some of those two regions that are activated when you inject the GLP-1 from the periphery, but still acting on the brain. So how to use this information? It turned out that GLP-1, we found out, didn't live very long in the organism. It was broken down within minutes. The half-life is 1 to 2 minutes. Crazy, crazy. So you can't use that as a medication. But we also found out why. That's because there is an enzyme called DPP-4 that cuts away the 2 N-terminal amino acids, as indicated by the red arrow up there. So this was interesting because if you inject, and that's what we tried, GLP-1 to people, and looked at how much survived in the intact form, you can see the little yellow curve down there. There's absolutely nothing. It's a waste of peptide. It doesn't work. The metabolite is shown in the other graph. So we said, oh, maybe if we inhibit that enzyme, that may work. So that's what we did. And here are some experiments that we did, again, on pigs. To the left, we're infusing the peptide to people, or to the pigs, I'm sorry. And you can see that the yellow curve, that's the intact, the active hormone. And even if we infuse it constantly, we cannot elevate the concentration very much. Then we could give an inhibitor, valent pyrrolidine, in the middle. And then after that, the peptide was completely protected. And then we could look at insulin secretion, and now it was 4-fold increased. So it worked. And that, of course, led the pharmaceutical industry. They caught up immediately on that idea. And they were able to do 24-hour lasting DPP-4 inhibitors. The most important one is sitagliptin, which is in the middle. Januvia was the name, particularly beneficial in combination with metformin. It was a tremendous success, and Bo Aron in Sweden showed that during a 1-year treatment, here we're looking at long-term glucose concentrations, hemoglobin A1c. In the controls, the green line, it rises, but in those that were treated with the DPP-4 inhibitors, it falls below 7, which was exactly the target in those days. And that became a tremendous success. And I'd like to emphasize that because this was one of the pharmaceutical advancements that came out of this, and millions of people all over the globe have been using this medication. But there was another problem. Here's the peptide again, and the DPP-4 in one end. But the other end is also a problem. That's because it's also extracted and destroyed in the kidneys. And that happens with a half-life of 4 to 5 minutes. So if you can solve the DPP-4 problem, you still have the problem with the kidneys. And this is what led to the development of the GLP-1 receptor agonists as we know them today. To the left, we have some that are derived from exenatide. That's a different story we'll forget about because they're not really good. But in the middle and to the right, we have the interesting ones. So what was happening was that the GLP-1 sequence was attached to a fatty acid. They did that at the Novo Nordisk company because they had already been producing insulins attaching a fatty acid, the Levemir, and they were almost a day-long acting. So they knew how to do this, and they created the liraglutide molecule, and it worked. Now we had a half-life of 12 hours rather than 2 minutes. That's quite a difference. Semaglutide was a secondary one, a different fatty acid, a different coupling, and it all of a sudden was 1 week in half-life. And that, of course, was extremely helpful. To the bottom, to the right, you have tirzepatide, which is also a GLP-1 agonist. It also has the same prolongation technology, a fatty acid, exactly the same, but it's also a little bit different. It has a slightly different composition. We won't go into the details of that right now, but it's very interesting. So how good are they? And this is what is shown here. How good are they? So this is hemoglobin A1C, the long-term glucose in a large group of people with type 2 diabetes that are being treated with tirzepatide. And what you can see from the top graph is that more than 50% end up after 40 weeks with an A1C of less than 5.7%. 5.7%, that is normal. You can get rid of the diabetes by using this therapy. They were mean enough to compare it to semaglutide, the blue curve up there, of the line, which didn't do quite as well. What else do we have of sensations? Now, here is the second sensation. This is semaglutide in a cardiovascular outcome trial and over 2 years. And what you are looking at here is major adverse cardiovascular events, death, myocardial infarction, stroke, things like that. And you can see that you could reduce that by 26% over this period of 2 years. Another, actually unexpected sensation. Here is, of course, today we already have a meta-analysis of all the cardiovascular outcome trials that have been performed, and here we have them all. And you can see that all that are to the left of the 1.0 line, they are positive, they work. The only two that doesn't work are the ones that are based on Exendin, probably because of pharmacokinetics. Forget about that. So the real ones, the good ones, they all work on cardiovascular safety and reduce the risk. What else does it do? So here we have stroke. This is semaglutide in people with diabetes, and a very significant reduction in the frequency of stroke during therapy in 2 years here. Again, a very important improvement because stroke is difficult. Now, this is dementia, and this was a very recent systematic review and a meta-analysis, and you can see that this was a group of 26 randomized controlled trials with 160,000 individuals in. 10 of them were with GLP-1 receptor agonists, and the other half was with SGLT2 inhibitors, another very important and good diabetes medicine, I must say. So overall, this group did not show any reduction in dementia, but if you looked at the GLP-1 receptors alone, there was a 45% reduction in the occurrence of dementia. And the SGLT2s, there was actually, not as significant, but an increase. So this is really interesting. It leads us to the question of Alzheimer. So there are observational studies out now in very large materials that suggest that there will be a reduced risk by 40 to 70% of Alzheimer's diagnosis in people with type 2 when they're treated with GLP-1 receptor agonists. There are 2 big controlled trials on their way. They will be finished around Christmas this year. They're called EVOKE trials. It will be extremely interesting to see if that still holds true. So another thing, I said this about the reward mechanism. This doesn't only apply to food intake, but also to opioid and alcohol use disorders. So here are observational studies on a very large number of patients because now it's almost 20% of the American population that has been on a GLP-1 agonist. That many. Okay, but here 500,000 and 800,000 with opioid abuse and alcohol abuse, and you can see that the reductions are about 50% in those that are taking the medication. So this works, and probably because of the reward mechanism inhibition. So kidney problems are among the most important, and this is a recent meta-analysis again from all the most prominent kidney people in the world, and they've come down with studied 11 trials with 85,000 individuals here, and there's a reduction by almost 20% of the people of the occurrence of progression of kidney disease or even improvement. So how does it do it? There are fantastic effects on a number of things, as I've alluded to, described, but let me also emphasize that there are actions on receptors on the blood vessels. And blood vessels is common for all the organs in the body, of course. And that can explain why this compound has effects on so many systems. And it also has effects on inflammation. So if you look at inflammatory markers, you can reduce these inflammatory markers very significantly. But of course, there is also the obesity thing, and obesity is in itself associated with a lot of complications, of course. So what happens? So this is semaglutide, and here we have an 18% weight loss over 68 weeks, and that changed the world that day. That was the change. And they also did a cardiovascular outcome trial in this population. 17,600 people were looked at for 4 years. And there was a reduction by 20% in cardiovascular mortality and complications. So this means that not only those with diabetes will benefit from this, but all those with the metabolic syndrome and obesity will also benefit from this. So what about the competitors? What about tirzepatide? Here is SURMOUNT 4 with tirzepatide, 88 weeks. The weight loss is here 26%. Really big weight loss. And even more importantly, the SURMOUNT 1 trial here. That's the first of these trials of tirzepatide in the high dose. Shows that it had a wonderful effect on body weight, but also it was followed for 3 years, and they were looking at the incidence of new diabetes, because the risk of obesity is, first of all, that you will develop diabetes. And what happened? It disappeared. A reduction by 94%. Amazing. So who will win? I don't know. But here are some of the trajectories of weight loss in time. And what I've put here are some of the new stuff. Right now we have the conclusion of the ADA, American Diabetes Association, and a lot of new stuff have been presented. And I plotted semaglutide on a high dose here, the triangle, and I put it on top of the curve for tirzepatide, and they're completely identical. So whether there is a big difference between them or not, I'm not sure. I've put amycretin, which is another one from the Novo Nordisk company, on top of retatrutide, which is a triple agonist, which is very highly talked about. It's exactly the same. So that is not the most important thing. So rather we should ask, 40% stay 1 year after they have been prescribed a GLP-1 receptor agonist. Why is that? Okay, lesson number 1 in pharmacology, everybody stops taking their medicine. Costs, you addressed that already. Very, very important. Let's do something about that. I don't know exactly what, but let's do something about it. And they will come down, I assure you. Some will stop when the desired weight loss has been obtained. If you don't do anything else, it's a bad idea. Some people do have the side effects, and the side effects will actually decide which of all these compounds will win, because we need to reduce the side effects. And then loss of reward. Oops. Perhaps it's not so fun to be on one of these medications if it takes away the pleasure of a good meal, the pleasure of a good glass of red wine, and it does, to some people at least. So that could be another explanation. And I'm saying this because, if you worry about half the globe to be on a GLP-1 receptor agonist, forget it, they won't. So what do? The most important that you can do is to use this period, which is up to 2 years of treatment, when you lose weight, to change your habits, your exercise habits and your food habits. And the good news is you can do it. Because of the weight loss and the anti-inflammatory actions, you will be better. You can move around. You can get up out of the chair. You can walk. And all it takes is a 30-minute walk every day. And you can change your diet, because you're not so interested anymore. You're not completely obsessed with what you put in your mouth. You can do it. You have your will back. So this is what is demonstrated in the graph here, that if you combine exercise with medication, then you can maintain the weight loss even after the termination of the therapy. So what I've said is that GLP-1 is an insulinotropic peptide. It inhibits glucagon secretion also. This is a double action. It inhibits food intake and appetite. It acts on the brain. And it shows that these are very effective in obesity and diabetes. And they improve the blood vessels and inhibit the reward system. They have protective effects on the cardiovascular system and offer renal and neuroprotection. And I've told you that, the best explanation for this is that they act on the circulation and on the inflammation. So there will be some busy years ahead for you here, because there will be a lot of new compounds that will all apply to the medicines agencies, not only in the United States, but also here in Europe, definitely. Thank you very much.
Many thanks, Jens, for this tour de force of GLP-1 discovery and some of the impacts it may have. I was about to ask you whether this is something all of us is going to take, because it seems to be the solution for every human illness. But I think you also gave us the answer that there are most likely a downside of the medal here in losing the reward. It doesn't sound that nice.
I think we have an obligation now to make sure that those people that have the metabolic syndrome, as we call it, abdominal obesity, problems with blood pressure, glucose, lipids, and all that, and that causes an increased risk of having cardiovascular complications, stroke, myocardial infarction, deaths. It's tripled. And those people you can treat. That's what I've been trying to tell you today. With these medications, you can actually do something about it. And that's why I think we have an obligation to give them that medication, to try to find out how do we disseminate this to as many people as possible. That will be the real job. I don't bother about everybody else, but these people that need it, they should have it, I think.
Yeah, and finding an appropriate way of using it, in not just giving medication, but as you showed in one of your last slides, using it as an aim to lifestyle changes, and therefore, maybe turning things.
Precisely, and I think it's very important that it stays a prescription medicine, because you need that instruction, you need the advice about the dietary intake. You will lose proteins because you lose weight. So you will need to modify your diet to incorporate that. So there are all these pieces of advice, you need them.
I have a question online for you, Jens. Has there been any signs of addiction to these medications?
No, there hasn't, and no, there hasn't been that.
Because that could be a little bit,
interesting. No, on the contrary, and as I indicated, the problem is to keep people on the medication rather. Yeah.
That's not good for the farmer, I see. Any questions in the room for Jens Jules Holst here? He will obviously be here for the rest of the day and being able to answer whatever you come up with.
Okay.
Otherwise, we'll thank you again, Jens. It's been a pleasure having you here. So we are moving on into the day, and after this speak, I'm not sure whether you will be interested in the sweets for the coffee, but at least we will turn to the coffee break now, which will happen outside here. And Your Majesty, there will be some colleagues meeting you and looking forward to discussing with you. So please, coffee is served. So please be seated.
Fernand is connected now, so it's okay.
So I hope you enjoyed the coffee break and the first part of today. We are moving in. I was about to say to more serious business, but I think the first part was definitely serious as well. And I got many nice feedbacks on that part of the program. So now we have a couple of presentations that are a little bit retrospective, looking back at the past 30 years, and then crescendoing up to hopefully polishing our crystal ball and looking into the future on what will happen. Our next speaker is Fernand Sauer, and Fernand is special. Unfortunately, he couldn't be here physically due to personal reasons, but he will be with us online, live. Fernand is both the mastermind about creating the European Medicines Agency, and he was also the first executive director here. So it's a pleasure for me to introducing Fernand, who will give us his reflection on the emergence of regulatory science and establishment of the European Agency. So thank you for being here, Fernand, and over to you.
Thank you very much, and hello to everybody. As you heard, I was unable to travel to Amsterdam due to health reasons, and I would have liked very much to meet colleagues and friends and visit the new site in Amsterdam. The title of my presentation is a little bit anachronic. Since 30 years ago, few people heard about regulatory science. The term only appeared a couple of years later. There is a saying, science doesn't rule the world, but certainly it has ruled the European Medicines Agency from the start and even before, during the preparatory steps. Regulatory science needs good regulations and strong regulators. I will address both aspects and mention a few people from a very long list of crucial actors. Next slide, please. The inauguration of the European Medicines Evaluation Agency, as it was called at the time, took place in London on the 26th of January 1995. And on this picture, you can see the chairman of the Human Committee, Jean-Michel Agnesson, the chair of the board, Tom Heppel, who is with us today, the chair of the Committee of Veterinary Medicines, Reinhard Kocher. You can feel that there was a relaxed atmosphere in the room, but you could hear the hammering next door because all the meeting rooms had not yet been fitted. And clearly from the beginning, there has been a real entente cordiale between the board, the human, and the vet part of the activities. And the scientific synergy of having human and veterinary products under one roof was real, starting with antimicrobial resistance. Next slide. The regulatory science concept emerged officially in corporate documents only in 2012, both in the work program and in several articles in the annual report. The prerequisite for regulatory science is, as I said already, sound regulation and smart regulators. But let's not forget that despite increasing transparency, the public trust in regulation and regulators remains fragile. with the spreading of anti-scientific view. I would like to add also that at present, citizens seem to be more concerned with shortages than with therapeutic innovation. And therefore, I think regulatory science couldn't be reserved to a scientific elite, but as far as possible, could be shared with civil society. And the whole development of the European Medicines Agency has shown the importance of patient expertise, research, scientific evaluation basis, and of course also in the daily healthcare. Next slide. The regulatory environment has changed a lot. On the left-hand side, you can see some serious issues in the 80s, like arbitrary decision, technically incompetent regulators, poor scientific debates. There were corruption scandals in certain countries. Few scientific officers in health ministries, mainly part-time university professors advised the ministers. Ministries had little scientific resources, and there were only a few agencies, such as the Bundesgesundheitsamt in Germany. There was also a predominant wait-and-see syndrome, people looking towards the FDA to know what should be the next step. And unfortunately, the FDA suffered at that time of what was called a drug lag. The scandals which existed in certain countries were unlikely to happen under the strict EU peer review. It would have been very difficult to corrupt so many experts from so many different countries. So I think since 1995, the existence of the European Medicines Agency and of the EU regulatory network more generally has remedies most of these shortcomings. Next slide. The first steps of EU regulatory science can be seen mainly in the 80s with the creation of the Scientific Committee for Human Medicine, CPMP, and for Veterinary Medicine, CVMP, the emergence of quality, safety, efficacy, and GMP guidelines, a common format for application, and a sort of informal codification. Under the chair of Léon Robert, I was asked to create the secretary to the CPMP, and then later, under the chairmanship of Jacques Boisselot, the secretary of the CVMP. I was then succeeded by Marie Dominic and Bob Hankin, respectively. Under these committees, in order not to stay idle, working parties were created. And we have with us today Rolf Bast, who chaired the Safety Working Party, and Jean-Louis Robert, son of Léon, who chaired the Quality Working Party. In the previous scientific presentation, I heard the name of Professor Whitberg, and if he is the same, Professor Whitberg was an eminent member of the Efficacy Working Party with Professor Alexandre, and he brought to us from the Northern Council of Medicine a very important good clinical practice guideline. The guideline, contrary to strict regulation, became then a tool of choice for regulatory science. Unlike regulation, it was flexible, easy to update, to put the state-of-the-art level. And it was also a pedagogical instrument between those who knew better and those who were still in the learning curve. Next slide. The evolution of the framework for regulatory science came in my opinion, 3 phases. The first phase from 1980 to 2000 saw the development of requirement. First, I headed a small pharma unit which produced 13 proposals for the 1985 white paper on the completion of the single market. The biotech procedure, which came in 86, prefigured on a voluntary basis the main EMA principle of functioning, and also introduced 10 years protection, which is still a controversial subject nowadays. In the 1990s, that was the start of the European Medicines Agency and of many national agencies. I would like to, since we have the presence of Petra Dor from EDQM in Strasbourg, I would like to underline the very important partnership with Agnès Artige and the Pharmacopoeia, which were very useful to take up the generic end of the market and biological standardization. Next slide. The second phase started with the codification of all the previous texts in 2001, and it led to constant improvements and a greater role for the European Medicines Agency, including crisis management in the recent years. The third phase will start probably in 2026 with the implementation of the reform, and mainly also with the impact of artificial intelligence, which may lead to a new pharmaceutical package in not such a distant future. Next slide. The EU nowadays can count on a solid EU regulatory network made up of the agency, EU Commission, and national agencies. The list of all the benefits is given here for 30 countries, including the countries from the European Economic Area. And the European system has been in constant dialogue with other agencies in the world. Start with the Food and Drug Administration, the ICH partner, the new African Medicines Agency, and the International Coalition of Medicine Regulatory Authority. Next slide. At the start, National Medicines Agency coordinated their activities, and from initial competition, mainly for having new substances going either centrally or to the decentralized system, from this initial competition, we succeeded to create a global partnership between the European Medicines Agency and national agency, with the support to decentralized activities. Here you can see the third meeting of the National Medicines Agency, where I was invited for the first time for the third meeting in Rome. Next slide. Starting in 1999, the European Medicines Agency invested a lot in the Pan-European Regulatory Forum, which allowed the candidate countries to participate in all scientific activities of the agency long before accession. And this started also when Norway, Iceland, and Liechtenstein were fully included in the European Medicines Agency operation. Next slide. I took this nice slide from a presentation done at the 20th anniversary with Anthony Humphreys and colleagues, which pays tribute to the hard work of all European Medicines Agency scientific bodies. And I hope that for this anniversary, this slide could be updated. When it comes to the European Medicines Agency governance, next slide, we have a picture here also at the 20th anniversary of the 7 board chairs at the time around Guido. And on the right-hand side, the successive executive directors, Thomas Lundgren, Andreas Pott, Guido, Rasi, with whom I entertained very pleasant exchanges. Next slide. I think one important asset of the whole system is the European Medicines Agency Permanent Secretariat, whose dedicated skilled members provide a lot of tasks which are listed here, hundreds of meetings, thousands of experts managed, scientific and administrative support, and the support for the Commission marketing organization. From 16 staff members at the inauguration, it grew to around 1,000 people nowadays. And I would like to reiterate the message of EMA, which she gave on the 1st May to her staff. And I could have used it at the very beginning. Keep being curious, keep asking questions. That is a duty of scientists in our society. It's true that the early recruitments were difficult. We got young, enthusiastic colleagues from all over Europe, but at the time, senior national staff were not so sure about the EMEA's future chances. So we started with an important learning phase with standard operating procedures and achieved fast results leading to a quality management culture. One of the premier worldwide, the European public assessment reports, was inspired at that time by Marisa Papadou. And the Secretariat was from the start the strict guardians of evaluation time, which was very important for our success. Next slide. The successful harmonization process was shared with other important partners in the world. We were able to recycle the internal harmonization efforts to reach out to other agencies and regional bodies, and with the WHO, of course, such as ICH. Here you can see the picture of the second steering committee of ICH in Tokyo in 1990. The Pharmacopoeia, to which the European Union adhered as a full member in 1994, and the VICH process, which started in Brussels in 1996. Next slide. I would like to pay tribute to good old friends, Noël Watillon from Fernand, who left us. Noël Watillon had a key role in developing regulatory science at the European Medicines Agency, and your conference room today has his name attached. Bert Farmer, first chair of the Committee of Orphan Medicine Product, was also able to welcome patient groups, not only for regular exchanges, but as full members of scientific committees and later even of the board. And I would like to meet here Yann, who will succeed me in my presentation, who was unanimously elected first vice chair of the committee, who represent patients and play a very active role in advancing their cause through EURORDIS. Next slide. A few pictures from celebration in 2010, 2015. In 2000, you can see Patrick de Boisert, who was my successor at the Pharma Unit, with José Aranda da Silva and Rui Ivo, first and present InpharMed heads. I was very lucky to participate in the inauguration of InpharMed and in the 30th anniversary in Lisbon as well. We had a celebration cake in 1915 with Christa Wirtmer and Guido Rasi. I didn't have any picture from what was probably a silent celebration in 2020 after the move to Amsterdam. I would like to underline the crucial role of head of agencies around EMA and their capacity to anticipate. The European Medicines Agency and the EU network of national agencies is an excellent example of successful combination of national and EU activities on very complex scientific issues over the last 30 years. No major failures were registered, no major complaints from patients, professionals, or industry. The European Medicines Agency has been able to anticipate many features of the future pharma package with pilot projects on COVID and also on shortages. And these pilot projects are facilitating greatly the discussion in Council and Parliament, leading to the final adoption of the package. I would like also to underline the international prestige of the European Medicines Agency and of the whole EU regulatory network. Next slide. The pharma reform opens a third cycle with many positive aspects, which I have listed here, also together with the Critical Medicines Act and the Future Biotech Act. The complex incentive regime of data protection is still a difficulty to be resolved by Council and Parliament. Another issue for me is really a question to DG Santé about the EU complexity. I mean, over time, the EU regulation has become very complex. And my two questions are, would there be an opportunity for regulatory simplification when the Commission implements all the new measures under the package once adopted? And my second question of simplification is, beyond the present reform, could the clinical trials regulation be revisited and simplified to attract more research in Europe? I will like to finish with the homage to the African Medicine Agency. There was a very recent encounter at the agency between African Medicine Agency and the European Medicine Agency directors and board members. I remember a very important harmonization meeting which took place in Accra in 2012. Guido was there for WHO, and Yves Juillet for EIA, was organizing the meeting. And Mimi Darko gave a very impressive presentation. One African colleague made a very funny remark, wondering whether this was all about PowerPoint harmonization, or would it become real in the near future? It has become real, although it took a long time. Thank you very much for your attention.
Many thanks, Fernand, for providing the picture, the bigger picture from going back into the 80s through the creation of the agency, and also pointing into a little bit into the future and also into the global perspective. You mentioned our next speaker in your talk, and I don't think we can, the reason our raison d'etre as a medicines agency is, of course, to approve medicines for the benefit of patients and public health. And therefore, patients are very central to our work, and it's a pleasure for me to introduce our next speaker, Yann. And Yann has been instrumental in creating the European Patient Advocacy EURORDIS. You've been the executive director for 25 years, stepped down last year. It's a really great honor to have you here, Yann. Please, the floor is yours.
Okay, I wanted to have a clock, but it doesn't work. So I'll do my best. Thank you, Stephen, and thank you, Fernand, for mentioning me and other colleagues along the way. First and foremost, dear colleagues, let me start by saying thank you. As a father, we wouldn't have his daughter of 35 years alive without your work. As a patient representative of 30 million people affected by rare disease in Europe, but also, if I may, today, exceptionally, on behalf of patients and consumers in Europe, thank you. Thank you to the European Medicine Agency, its directors, its board members, its staff, the thousands of experts involved, the national agencies, the European Commission, and the member states' regulators. Thanks to you, you enabled science to translate into innovative human medicinal products, treatments, and vaccines, transforming public health, transforming healthcare for all citizens in the EU, transforming our life. We can be all proud of it, and maybe today is a moment of pride. My talk will be in two parts: the benefit from patient perspective and the engagement of patients. First, the population at large and patients have benefited from these 30 years of European regulatory framework. The body of the EU legislations and policies has enhanced the collaboration between the member states' regulators, but also between regulators and stakeholders at large. Companies, scientists, clinicians, patients, consumers, healthcare professionals. We, as patients, as population, we directly benefit from a European common scientific advice, from a European common benefit risk assessment, from a European common pharmacovigilance, and much more. Collaborating at the European level has enabled to work with the best experts in Europe, whatever their nationality, and to pool their expertise to our benefit. Thanks to this European framework, we also all benefit from a more robust scientific ethical development and assessment, and overall from a common culture of collaboration, of quality, and evidence-based decision, in particular focusing on clinical evidence. and pursuing both excellence and public interest. The European regulatory framework and the EMA, I would argue, are a high-value public good. It brings more equality to the citizens in Europe. In its absence, we would have more inequalities of expertise, of assessments, of decisions, and availability across the territories of EU. And my second message on that part of the benefits is that looking back at the 30 years, patients in Europe have benefited from legislations and policies targeted at specific populations. The regulatory on orphan medicinal products, 1999, the regulatory of pediatric use of medicines, 2006, and I would say also the regulation on advanced therapy medicines in 2007. These three regulations have promoted the European Commission and EMA at the forefront of the two world leaders with the FDA. The regulations have enabled to address high unmet medical need, and I'm pleased to see that ambition in the new revision of the pharmaceutical legislation. It has enabled to steer scientific and medical advancement, to attract both public and private investment in the development of therapies, to build clinical research infrastructures, which is so critical for the coming years for attractiveness and competitiveness, and to develop new clinical trial design and statistical methods. And overall, these regulations of specific populations have core elements of the European ecosystem for clinical trials and biotech. So while we are preparing the Biotech Act and revising potentially the clinical directive, we should pay attention to the specific regulations. They are building blocks benefiting to health needs beyond the rare and the pediatric, and also health priorities at large. Through these regulations targeted to specific population or society, meaning EU and member states, provide additional efforts to people with special needs who are more vulnerable in our society. By doing so, they create more equity. When Europe delivers more equality and better equity, driven by public interest, it delivers on its hope. And there are important secondary aspects coming along the way. This European regulatory framework not only built the great European agency that we are proud of, the EMA, but also builds a resilient, decentralized network of national agencies with the head of medicine agencies mentioned by Fernand a moment ago, making each national regulatory agency stronger, particularly in the specific population, I would argue, and I hope they will not lose that with the new pharmaceutical legislation, and offering inspiring model for Africa, for instance, like was just mentioned. Because the European regulatory framework is anchored into the European market integration, it is a powerful steer for European scientific collaboration, for converging member states' health research agenda, and for a growing EU health research program and EU public health program, and I hope this will continue in the next MFF. Overall, more research, more infrastructures, more clinical trials, more competences, more investment, a culture of collaboration and excellence. And in fine, more medicines, as you mentioned, Emer, over 400 approved at the EU level in a more robust, reliable, predictable system, also more fair, and much better than if it had been fragmented in 27 national frameworks. So when Europe delivers meaningful progress to its citizens, it delivers on its hope. But I also have a message of concerns. You know me, I cannot escape that, right? Concerns when looking at the next decade. First, the European Union lacks strategic alignment based on the long-term and global vision. Too many, if not most, of our legislations and policies over the last 30 years and even in the last 10 or 15 years have been developed with specific goals at specific moments, which is great and we can understand. But piling up legislations and creating silos with inconsistencies and inefficiencies, overall, we're losing opportunities. I hope that the new strong momentum for EU competitiveness and attractiveness at the global level, with an alignment at this moment between the Commission and the European Parliament, and hopefully a majority of member states will address this vital issue. The revision of the pharmaceutical legislation, together with the Critical Medicine Act, the Life Science Strategy, and the Biotech Act, are a great opportunity. But there is also the possible revision of the diagnostic legislation, of the Clinical Trial Directive, and then the Cross-Border Directive. And I know it's a lot of work to swallow in only 5 years, but we need that alignment in order to reach the strategic outcome that we're looking for. The second is the initial ambition that all patients in the EU could benefit as equitably as possible from the very best treatment. It's still not a reality. Still not a reality. The last 30 years have not sufficiently addressed the slow and unequal and illegal access to patients for patients to medicines in Europe. We know why. We had plenty of work and assessment and conferences around that and work. We know what to do, and we know it is difficult. But this is paramount for the next years. Competitiveness is not only about bringing this strategy together, it's about, at the end, making sure that every patient has access where it lives at the right moment to the medicines. Europe needs to do that to show that it delivers health for its citizens. Streamlining also regulatory and HTA advice and assessment, and improving decision-making on pricing and reimbursement through more collaboration at the European level is essential for the innovative therapies of tomorrow. It will not work through the fragmentation of 27 member states. It will be an announced disaster, particularly for the most advanced therapies and the personalized medicines. Lots of progress are being made. I know the efforts, and that's fantastic. But we need to go faster and further, and with the proper funding for this agency and also for the European HTA in order to have the proper collaboration, but also collaboration with payers, or I would prefer to call them the buyers, as being critical. And my third message on looking forward is really some specific population, if I can call it like that, which are still forgotten. Women health, and I'm pleased there was an intervention later today on that. Women health. I mean, we're discussing that since so much time. What are we doing to address the specific needs? And I will add aging population. We're talking about the pediatrics. Good, but what about the aging use of medicines? My second part will be faster on patient engagement. The European regulatory framework and EMA have pioneered, I would really argue, you have pioneered stakeholder engagement and active participation of patients' representatives into the scientific committees and into the EMA scientific procedures and the EMA management board. The first meeting of patients goes back to 1996 between the CHMP and an AIDS advocate for meeting on HIV, and that was followed in 97, 98, 99, and if I may mention, François Houillet, my former colleagues from EURORDIS, being the lead advocate with colleagues, but also Jean-Michel Alexandre, who was mentioned, Fernand Sauer. It was thanks to them that this was made possible, and opening the door then to a change in the regulation with the orphan drug regulation. And that came in 2000, when the first 3 patient representatives were appointed, including myself, and then having elections as vice chair. It was not a given. We had 6-1, 6-2, I think, something like that. What I'm proud of is that 3 years later it was unanimous. So it shows that there is some value, some time that we participate, right? And I'd like to mention Josep Torrent-Farnell, the first chairman of this committee, and all the chairmen of this committee, including Bruno and Violeta, who are here today, who are fantastic chairmen, but also all the patient advocates over the years who are contributing their personal time and efforts with huge work. Today, there is patient representative, as you may know, all of you, pediatric committee, in the Committee of Advanced Therapies, in the Pharmacovigilance Committee, and have patients as full members and voting rights. But it's not easy, you know. It's a lot of work, and you don't get much support to do it. And it's important to be more than one in the room. And each year, there are now over 1,000 participation of patients in scientific committees, in scientific procedures, working group in the EMA, and I would like to say thank you for that too. Scientific publications, with EMA being a co-author, shows that there is a scientific value of having patient involvement, having an impact on the scientific outcome. The first meeting in 2002, 2003 of patients' representatives here was organized to form the Patient Consumer Working Group, which became, a few years later, the Working Party of Patients and Consumers. And the regretté Noël Vation was behind that with Patrick Lecourtois, which may be with us today, and Nikos Dedes as the first chairman. In doing so, the EMA has been ahead, and I would like to insist on that, ahead of the FDA. FDA has created the very interesting patient focus group, and I think we should take lessons of that. There is some very good outcome of that. But also the patient engagement committee of the FDA is patterned on the experience of EMA, but is not delivering as good. And FDA has more public hearing, yes, but you have a lot of lobbying around these hearings. But EMA, what the EMA does better and more than the FDA, and should claim it more, I would argue. Stakeholder engagement over the years has brought quality information, transparency, prevention of potential competing interests, and public debates. EMA and the European regulatory framework are trusted. Even in crises like the reintroduction of thalidomide, it was trusted thanks to the intervention of patient representatives as diplomats into that effort. Trust and name recognition has increased with the COVID crisis, but trust cannot be taken for granted, as Fernand mentioned. Social media, fake news, devalorization of institutions and of science are real risks. And EMA is taking the very good steps in that direction to try to address these challenges, and the network of stakeholders engagement is a crucial asset. My last message in looking forward and conclusion is that there is the ongoing pharma legislation review, and as patient organization, we do support this new legislation. But the patient representation and civil society may be strongly undermined during the trilogue. The European Commission proposal is good, and proposing 4 patient representatives and 4 alternate with voting rights. European Parliament is supporting having them, these 4 representatives, in the CHMP and in the PRAC, in the Pharmacovigilance Committee. But some member states are reluctant and opposing for now. So I encourage you to reach to your representative of member states in the European relevant group supporting the Council and the trilogue and advocate to have the patients there. And why that? Because meaningful patient engagement with voting rights is important to be real and credible, not just witness and actors into committees, but to be colleagues engaged in a co-work with the other colleagues from the agencies, to have the patient organizations and patient reps to act and work in a responsible manner. That's important. If you want them in the next 20 years to play in a responsible manner as individuals and as organizations, give them the same responsibilities. And to enhance the trust of the public, to bring the democratic process, transparency and trust at the core of the expertise. I hope the member states will be wise to enable the European Medicine Agency to continue to lead in that regard in the world. Thank you very much.
Many thanks, Yann, for this very clear and strong message. Well received, many thanks. We are moving on. The next speaker is Guido Rasi, and the reason for picking Guido is that he was the executive director here from 2011 to 20, when the agency was undergoing a lot of challenges, moving out of the UK, and then suddenly Brexit came. No, not Brexit, COVID as well. Brexit and COVID. So the floor is yours to tell us about leading the agency in troubled waters.
Yeah, thank you. Thank you, Stephen. Thank you, Emer. Thank you for organizing this. Thank you for inviting me. Thank you, Stephen, for involving me in the program. And yes, I will give a speech not much on management, but more on people. As you know, we all know how EMA works. It works in the innovation domain, which means that you have to find continuously solutions, innovative solutions, creative solutions, sometimes in a very fast evolving scientific and technological environment. And it has some peculiarity. EMA operates under the public administration rule, and rightly so, but with a need for a private level flexibility. It's providing service to public health, but to private companies, and it's also benefiting a lot from experts from the European network, but it's also depending from that. So meanwhile, it is balancing these elements. Sometimes environmental things happen. When I took over, there was a transparency issue. We have been depicted like this. The Mediator scandal was floating around, and we were under attack in the media. 2 discharges denied in a row. Internal, long and strong debate whether to take courage and make it public what we use, the data we use to take our decision, with a lot of concern of possible legal consequences. The internal discussion ended up that we all agreed to be brave and go for publication. And I remember that we all agreed that I was cast, and I pronounced the famous, I see someone who inspired me. It's not a question of if, it is a question of when. And the next year we have been depicted a little better. Things improved a little bit. This became real 2014, January 2014, and we enjoy to be the first regulators in the world to publish the data we use to take our decision. So, with this restored reputation, never denied a discharge so far anymore, back to business as usual, addressing new paradigm shift, scientific discussion, implemented the pharmacovigilance, the PRAC was established. Patrick Lecourtois was retired, so we need to readjust the balance of the 2 kingdoms that were inside the agency. And we went to review and reconnect. And the first building relocation is to be continued this story. The first relocation, and but the 30th of November 2014, a judgment of the Civil Service Tribunal annulled my appointment based on a bizarre interpretation of the conflict of interest of the selection panel, not of the candidates. And it was a difficult time because the agency was also challenged about the validity of 3 years decision that I signed off. And we convened all together, extraordinary executive board meeting, management board meeting, general assembly. Reaction of people has been really, really exceptional. People was completely devoted to the agency task, putting aside any personal problem. I felt completely supported in my transitional role of strategic advisor to the acting executive director, Andrea Spott, who was simply loyal, fair, and efficient. At time, sometime, is a gentleman and paid me back. During the year of the selection procedure, the retirement law changed, and I enjoyed to have a full mandate eventually, which never would happen without that wrong judgment. Well, so after that, back again to business as usual. The international arena was taking the stage, ICH was high, ICTRA, establishment of ICMRA, increasing worldwide quality issues, perceived conflict of interest in hiring and recruiting new experts and new expertise, reorganization continued. I had an attitude to reorganize. The first joint EMA-HMA strategy was kicked off, a really important step. Shortages management started to be a problem. Patient access to innovation was high in the discussion. Health technology assessment was taking a lot of time and evidence, and was the start of real-world evidence, the prime scheme, a lot of good things and difficult things going on. But the 24th of June 2016, in few hours after the decision of Brexit has taken, an ad hoc General Assembly has been convened, as you can see from the participation. We have been really lost. Will we have a new home, a new house, where we will be going? Immediately a business continuity plan, an ad hoc team, management and staff, continuous involvement, continuous consultation went on. But listening the proposal from the EU Environment and Media, we had a lot of concerns, a lot of doubts. So we focused on the strategy for the relocation and was developed putting up front real elements: staff retention, transport, school, housing, analysis of potential critical capacity due to expert and expertise loss. So we took a rational approach versus an emotional geopolitical nonsense. And we took an historical staff survey. Was held, made public. Not many people happy, but it worked. After that, we had to say goodbye to London, flag lowering ceremony, emotion, sadness, feeling of loss, but also strong feeling of belonging, and we were all singing together the EU anthem. And 2017, anthem was decided that the new home and house, the new host country for EMA, in March 2019, with a lot of enthusiasm, you can see, we were ready to start again. Relocation number 2. And relocation number 3 soon after, in January 2020. Welcome to EMAB, this building actually, with flags up again, with enthusiasm and commitment as usual. How was possible for EMA to survive and to be successful? Was resilience of people, was the sense of mission, of purpose, and was the social impact we are aware. Sorry to say we, but I feel still belong. The social impact that is very high in this agency. So seriously back at business. The future is bright, everything is known. Long-term project, succession planning, a new executive director. Now we have EMA, but that was the time that I was going to complete my mandate. New staff hiring, new medicine generation to address European surveillance, veterinary, antimicrobial consumption to further support. Data were coming, but COVID pandemic, which means empty rooms, first management board and CHMP online, a lot of things going online. People involvement, again, everything was kind of known this time. And just a reflection before I go to conclusion, how it happened that overall EMA was resilient and performed all the time through this. Was the governance structure that helped us, so we always strive to find, to have an automatic governance giving us the perfect balance between processes and procedure versus flexibility, compliance versus delivery, control and monitoring versus delegation and trust, hierarchy versus leadership. We strive to have the perfect organization. I tried, but I learned a very simple lesson at the end. A perfect orthodox structure with the wrong people attitude will fail. Meanwhile, an average designed governance, we need a governance, with committed people will perform beyond any expectation. So, what is the common element? We have 4 examples I gave. Reputation. Yes, we were driven by transparency, follow the principle. This was happening inside with anytime with involvement of people, tough discussion with strong personality, believe me. But at the end, we come to a decision, one more consultation, take the decision, don't drag it. And people involvement was the key. The wrong civil tribunal, awareness, resilience, support from people. Brexit, evidence of the need, respect for people's lives versus geopolitical emotion. And COVID, experience, sense of duty and belonging. And experience, I can tell you, we were convening some meeting with the agenda of the day in front. We are looking in the eyes, closing the agenda, walking out, we knew what to do. Very well trained. And one more element, if you have some founder that put the agency on track, Fernand, brilliant enough to make in a good spirit another colleague, you can see Noel, how happy was. Well, believe me, having outstanding partners, it's helping you to be a better manager, by sure. And thank you to my best and continuous partner. And people is making the difference. And thank you for making it possible. Thank you.
Many thanks, Guido, and especially for highlighting the most important asset of this house, the people who are in it, the staff. Thank you. Really, really excellent. And now we had 3 talks focusing a little bit on the past and the recent past and the challenges. So now we are going to think about the future. And as you know, prediction is difficult, especially about the future, but I'm pretty sure I found the right person for this, María Lamas, the head of the Spanish agency, but also the Heads of Medicine's management group chair at the moment. And María, you promised me to talk about imagining the future of science and regulation. So please, María.
Thank you, Stefan. Thank you, Emer, for inviting us to participate in this celebration, this joyful day. It's incredible for me to be here today in front of you. Stefan, it was not exactly that I promised to speak about the future. You proposed me to speak about how I imagine the future. But I have to thank you for doing so because I couldn't like it better. Which is better than imagine the future? That means dream of the future. So that's not an easy task. 30 years ago, Stephen Hawking said the world is changing faster and faster, so we can expect big changes, some very exciting, some alarming. The one thing I am sure of is that it will be very different and probably not as we expected. And I can't get over it. That's, I think we can repeat what he said. But even though we have a mission to accomplish, and I think that overall The mission of the whole network, regulatory network, is to promote and protect the health of humans and animals through medicines based on scientific excellence. And with this in our mind, I invite you to dream of the future we aspire. For example, I would like to see that children that are born with rare diseases have real expectations of a healthy and functional life. I would like that infections are under control with successful and preemptive measures, including vaccines, and the use of effective and safe antibiotics when they are needed. I would like to see that oncological diseases are controlled thanks to good early diagnosis and also to the use of effective medicines that when they are needed, they are perfectly compatible with normal life because they are perfectly tolerated. I can imagine that cardiovascular diseases are a thing of the past in most cases, thanks to the excellent preemptive way of life, healthy lifestyle, as Professor told us before in her keynote speech, but also including the use of medicines to mitigate the effect of these diseases. I also dream of autoimmune immune diseases are controlled with excellent medicines that keeps patients free from symptoms or outbreaks, or that the elderly have a healthy last stage of life with a neurological status that allows them to keep their functionality and autonomy. And I could add many other dreams, but one of the basics for this make these dreams real is that everything will be supported by a rigorous, reliable and competent European regulatory system led by the EMA. But we regulators, that we have a scientific background and we are scientists regulators, we prefer to shape the future rather than to imagine the future. And organizations have a very useful tool to shape the future. It's the strategic planning. And for that, we deliver every 4 years, more or less, our strategic plan. And we have recently presented our EMAS 2028. This is the first step towards the future, only to 2028. But I think we are in the good direction. In this plan, we selected 6 strategic focus areas, and I invite you to go over these 6 areas. The first one is accessibility. Our vision, our dream, is that European patients have access to the medicines they need in a timely manner and no matter where they live or what economical status they have. And we dream of this, including the feature of affordability, in spite of recognizing that affordability is not part of our remit, but we don't live in an ivory tower, and we know that we can collaborate to make medicines more affordable and accessible to all European citizens. And for that, we will work, and these are the goals within our strategy. We will collaborate to have an effective HTA regulation implementation, to foster a robust scientific evidence that serves to all the decision makers. We need the same criteria for to be accepted by HTA decision makers or by regulators that assess medicines for granting authorization. We have to be sure that all the medicines, breakthroughs, but also patent medicines, cover all the patients' needs, even unmet medical needs. And we have some examples of how to make this real. You can consult through the QR code. The new EU rules for health technology assessments become effective, displayed on the EMA webpage. The second strategic area is leveraging data, digitalization, and artificial intelligence. And our vision here is that the development of medicines will be faster, procedures are agile, the medicine world is interconnected, data generate impactful evidence. And our strategy sets at least 3 goals, like maximizing data utilization and interconnectivity, interoperability, And for that, we foresee to embed EU healthcare data in the network processes, to pilot novel data types like synthetic data, omics, prompts, to ensure, keep the guarantees on the data quality and the interoperability. A second goal will be to leverage digitalization and experimentation in order to optimize regulatory processes and to strengthen digital infrastructure, and also to progress on the veterinary medicines digitalization. And the third goal is to make real the AI's potential, both in the throughout the business, the whole, to support the digital business transformation, but also focused on the medicines, covering the whole medicines life cycle, from the early research, clinical research, assessment, and vigilance after marketing. The third strategic area is about regulatory science, innovation, and competitiveness, although competitiveness is embedded in the whole strategic plan. And here our vision is that regulatory science integrate all the scientific and technological breakthroughs for the benefit of patients, human and animals, while respecting the environment and avoiding the use of animals for experimentation. We aspire that ambitious clinical developments bring medicines to cure or alleviate the currently unmet or unsuccessfully met needs. This means that we expect a significant magnitude of benefit, not only a clinically significant benefit, also a vitally significant benefit, because we aspire to change the lives of those who suffer the most. And for that, the strategy sets 3 goals, integrating the advanced science in medicines development and manufacturing. We are seeing great progress in new novel manufacturing systems. We will work to foster innovation of impactful evidence. This means to leverage the power of digitalization, but this means also to set evidence patient-driven, and also to aspire to maximize the benefit achieved by the new medicines. And for that, we will support a close cooperation with the stakeholders, with academia, with researchers, with industry, with patients, to accelerate the translation of innovation. For achieving these goals, the work of one of the working groups within the network, the EU Innovation Network, is really essential, pivotal. You can consult the work plan through the QR code. And one of the exercises in charge they are taking care of is the horizon scanning reports. Horizon scanning are a very powerful tool to scrutinize the future, trying to guess what's coming up. Not only to be prepared for that, but to be part of the development of the way towards the future. So in this, the example that I am presenting in the slide is the last horizon scanning report. It's about new approach methodologies. And the aim of this report is to identify those methodologies that progressively can incorporate alternative methods to, for the preclinical development of medicines to accomplish the 3 R's principles: replace, reduce, and refine animal models. And they identified several methods: in silico, in vitro, ex vivo, in chemical approaches. And if you want more information, it's nice to see how this bibliometric network analysis have identified a lot of research projects, papers with novel approaches in clusters, classified in clusters, for example, with the cluster of molecular docking, or machine learning, or tissue engineering, organoids. So there are a lot of information in this report that make us be more confident in that future without the use of animals for experimentation, for example. Another important action within this strategic area is to foster the clinical trials in Europe. And for that, we are part, the EMA, along with the national agencies, their network, HMA, And the European Commission will lead this initiative, Accelerating Clinical Trials in the EU, ACT-EU. And the objective of this initiative is to transform the European Union into a region that supports clinical trial development and enables collaboration and innovation in all stages of the clinical research life cycle. But in a nutshell, what we want is to make the European Union the leading region in the world by 2030 in clinical research. And for that, we are analyzing, trying to incorporate the new methodologies of clinical trials while ensuring the integrity of data, the protection of patients and participants in clinical trials. We engage with academia and industry. We work closely with them in the platform, with the stakeholder platform, to receive the direct feedback from them, because that's what drives our actions. And all the system of clinical evidence is patient-driven. Questions are posed by patient needs, and evidence is built on the previous evidence generated. The fourth strategic area is about antimicrobial resistance and other health threats. And our vision here is that infections, as I said before, are under control with successful preemptive measures, including vaccines, and the use of effective and safe antibiotics or antimicrobials when they are needed. And for that, the strategy identifies three goals. The first of them is to contribute to the responsible use of antimicrobials, fostering the stewardship programs. And for that, we will increase our efforts in the categorizing and collecting data of consumption of antibiotics both from human and veterinary use. We collect from veterinary use, but we use both sources to modernize and update the product information, especially when it comes to veterinary antibiotics, and to develop regulatory guidance on antimicrobial use in animals. But here, when we are talking about development of antibiotics, this is a known goal, because we know that there is a clear market failure with antimicrobials and antibiotics. So one of our goals is to support development of these new antimicrobials, keeping systematic support to developers and facilitated early discussions with them. But we can't forget that antimicrobial resistance is not the only health threat we are facing. We have very fresh memories of the last pandemic, and we know that we have to strengthen our preparedness for future health threats with One Health perspective, and taking into account that environmental threats are a new important threat. We are seeing how vectors are the displacement of vectors from southern regions to northern regions in Europe. This brings new zoonoses, new potential infectious diseases. So we have to be prepared for new diseases that we haven't had in the past in our region. We need to be prepared in case of an emergency to support end-to-end the development of medical countermeasures, and also to set an appropriate system for timely clinical trials. And all of this with the necessary flexibilities that Guido remembered us how we did in the past pandemic. The fifth strategic area is about availability and supply of medicines. And our vision here is that we will have all the medicines we need, because we will count on a reliable supply chain, including veterinary medicines. And for that, the strategy sets two goals: strengthen medicine availability. We have worked very hard in this, and also to reinforce supply chain oversight and increase inspector capacity. And this is really important, because we see from innovation that we will be facing new novel manufacturing systems, methods. We have to update our GMPs. We have to be sure that our inspectorates are ready to carry out these inspections with these new criteria. We have enough capacity and capability to cover all the manufacturing and this delivery supplies chain. But also in face of the next Critical Medicines Act, We will also have, I say here, the technical criteria for supporting the selection of critical medicines will come from the work of regulators. And the last strategic area, for me, it's the most essential, sustainability of the network, because without the resources, the adequate and proper resources, we won't be able to achieve the previous goals. So the network, our vision is that the network will have the necessary resources, capabilities, and operational model to effectively pursue its long-term goals in protecting public health. And for that, our current strategy sets 3 goals: reinforce scientific and regulatory capacity and capability, to establish a shared operating model to support network activities and collaboration. And for that, it's essential, and I invite you to consult the work plan by the Network Data Steering Group, including actions on plans about the use of data, interoperability, and uptake of AI in the medicines regulation. And also, last but not least, to strengthen public and stakeholder engagement in defining and designing how we should operate. And my last words are for those who make this possible, for regulators, for the former regulators that have worked for decades so far to bring us the current high-level standards of medicines, for the current scientific regulators that are actively working in providing a more consistent, efficient, innovative, and patient-driven regulatory science to ensure the quality, efficacy, and safety of medicines, but also they are also shaping the future. And also a word for the future regulators, those that in 30 years, some of them can be sit here, can be following the event online. Some of them can be in the nursery or even to be born. But these future regulators in 30 years will have made our visions real. So to them, my deepest recognition and gratitude, the past, the current, and the future regulators. And with that, congratulations to EMA on your 30th anniversary.
Many thanks, María, for moving us into the near future with the strategy to 2028. I know we are running a little bit behind time. I want to not steal 15 minutes of your lunch break, but postpone your lunch break with 15 minutes, and not stealing it, so we just postpone it. So I'll invite Emer, Guido, and Yann to come up here for a quick discussion, because we're going to discuss what could potentially be disruptive for our future plans. Okay. So we put together, I think some of us are going to be a little bit hidden for the audience, but I think we can fix things here. Can we move it? You just move this one? That's okay. We didn't know that. So my first question to you, and I hope you can all give a very brief impulse statement here, and I'll also put the Slido QA up so the audience have an opportunity to present questions to us. What do you consider the potentially most disruptive technology or event? I think I'm going to say event, so that could be technology, it could be political, scientifically, that would have a major impact on our future as regulators, but also the future of drug development. And I'll start turning to my boss, Emer.
Well, thank you very much, Stefan. And I think we've heard some wonderful dreams, past and future demands, and actually it's all about the people. I think these have been very consistent messages. If I look at what I think are the disruptors, the things that we need to take into account if we're going to imagine the future of regulation, I would focus on three things. Artificial intelligence, data, and then I wanted to add geopolitics. Not really because of geopolitics, but because of trust and trust in science, and I see them quite related. So first of all, I think we have a huge responsibility to really use artificial intelligence in the best possible way to make our lives better so that products can reach patients better. And we have to do it responsibly. And that's a challenge. It's a disruptor, but it's an opportunity. Data. If data is the new oil, we need to know how to make it flow. I think we're at the cusp of an amazing innovation in Europe now with the European health data space. This, for the first time, allows us access to electronic health records. And we can learn so much about that, but we have to do it well. It will disrupt the way we work. It will make us think differently. And then, of course, we never know what's around the corner. And I think this is one thing that I've learned from my 30 years. We never know what's around the corner. So as soon as you think you've got a plan or a strategy, something changes. So you have to expect the unexpected. And here it is about how we use the current geopolitical context to ensure that trust in science remains paramount.
Thank you, EMA. Yann, can I turn to you?
Yes, so I will mention also.
We need to turn it on.
Okay, super. Thanks. I will mention also AI with its exponential capacities and field of uses. Just to mention two examples relevant for us, the use of AI obviously in clinical trials all along the life cycle, but also to identify more repurposing of medicines or of assets which have been abandoned. The example also of the use of AI to generate patient preferences, then being curated by patient advocate. And there is a pilot called Collaborare between the European Medicines Agency and EURORDIS, for instance, in the field of rare diseases. The second I'd like to mention is scientific advancement, but focusing particularly on the extremely rare disease. So the 4,000 rare diseases, which are this long tail of diseases affecting less than 1 in a million in the population. And here, obviously, we can think of many technologies coming up, the mRNA, the CAR-T therapy, etc. But there is one that I would like to single out, which is gene editing. And sickle cell anemia, obviously, is now has gene editing approved. But there is also a recent outcome breakthrough in the US on a very rare disease called CPS1 deficiency, which shows that there is shortcuts that can be taken on all the development process, something that was taking before 7-9 years, taking less than a year, and requiring here a lot of flexibility, adaptation, while keeping the robustness all along. And this type of technology is cheaper, maybe really transformative. So I'm mentioning this one, but I'm sure others will come in, and we need to see how we will adapt as a regulatory system also to this treatment, which is going to be not more than personalized, individualized, or for very few patients. And here, obviously, the European added value will be maximum one time again.
Well, difficult to add things because what Emer and Yann said are, of course, I agree. To try to be very short and specific answering your question, what will be the most disruptive thing? I think will be inertia. The most disruptive will be inertia because, as Emer said, you don't know what is next, but we know that already on the horizon there are a few things. So if Europe, I'm talking about Europe, world is too big for me.
That's how it is.
And I'm aging. For Europe will be something will be left from US, something will be taken from others, mostly in Asia, science, the so-called Western science will be taken by others. Meanwhile, we are challenging our own science and history. Others will benefit of this, China mostly. So the only real disruptive thing in a negative way for us will be inertia. If we don't act fast, coordinated, now, we abandon the liturgy that we have to take decision in Europe, that will be really disruptive.
Thank you. I think that we are facing many potential disruptive features around us, and not all of them come from the science and the technology, also from the society. And I think that we are living in a moment where the expectations on the relationship between society and institutions is changing. And we have to bear in mind this, because we can't take for granted the confidence in science or the reliance on institutions. And we have to win them over every day. Building confidence takes time, but losing confidence is something sharp and acute. So we have not been so challenged ever before, I think. And this will change, and we have to adapt to this continuous scrutiny and to optimize how we connect with the society and communicate our work. But looking at the scientific side, the obvious reason is everything related with data, because we are fascinated by data and artificial intelligence. So I guess clinical evidence and the way we generate clinical evidence since the very beginning, since the preclinical and also the clinical research will change sharply. But not only changes will come from data. I am seeing very inspiring and promising advances in the manufacturing setting. Just an example, 3 days ago, Nature Chemistry published a report, probably you have seen it referenced in the media, where some authors and chemists were able to transform PET bottles into paracetamol using an engineered metabolism via Escherichia coli. So we are seeing things unimaginable a few years ago, or genome editing. Less than 1 month ago, an American hospital published the first case of in vivo editing genome to treat an inborn with a rare disease affecting the cycle of urea, and how this editor, vehicled by a nanoparticle, was able to decrease the number of hyperammonia crises, and for a disease with more than 50% of lethality, the patient is under control. So I think we are going to see from genome editing things that we can only imagine. And for novel manufacturing, also very promising advances.
So many of you mentioned AI. I think that was the hottest topic, and I had a pre-call with all of you, and I think none of you were speaking against AI as something that was disruptive. Also, the distrust in science is a big challenge, and I actually have a quite interesting question here online, which covers those two areas. Could AI work against the best interest of patients and science by making public unsure what is true and what isn't true? Do we have a chat? And what could we as regulators do there, if we can do anything? Who wants to pick that up?
I think Yann should. But it's about the patients. I think one of our challenges will always be how we reconcile what we do with the eye with patient trust, and how we ensure that we keep the safeguards in place. Because anything is possible. I think anything is possible. But we are medicines regulators, and our job is to do the right thing, Now, I always say this, and people look at me and say, well, what is the right thing? And sometimes we don't know what the right thing is. But together, I think we will figure it out. So I think there are challenges. I think we're right to have concerns. I think we're right to look at everything. But if we don't do something, and I go back to Guido's point, inertia is worse.
I agree, and in fact, I mentioned it rapidly in my speech earlier. This trust issue is, as you said, extremely. It's important, but it's very fragile, and I don't think it's a topic for the future. It's already a topic of today, and EMA is already working on social networks, for instance. But we can see that here also there is a conjunction between technologies, yes, but also the values in society and the political level. Look at the US, how in only a few weeks the FDA has been undermined, and how the panel of scientists on vaccine has been changed in order to give a certain direction. So just for the pleasure of the discussion with Guido here, I agree with the inertia. I think we do share that and that concern and the need for strategic alignment. On the other hand, what I observe is that the structure of Europe somehow offers less political exposure in the changes like in the US, and that provides maybe some resilience and robustness and predictability in a system that maybe we can take pride of and play more with than what we see today in the US. We cannot be happy about what's happening in the US. I think we're all very sad and concerned, but I see the opportunities for Europe in a moment where we've lost some time, some leadership. And thanks to colleagues like yourself, Madam, playing a lead role on the EU Clinical Trial Act EU and the European Medicine Agency network, that's ideal. That's exactly what we need to seize that moment, to really work strongly and without any preconceived ideas to make Europe more attractive and competitive on clinical trials, because that's the game for the future and that's what the patients really need.
No, quick comment, short answer to AI can be possibly detrimental for patient, of course, as any other novelty. Can be good or bad as long as you use for good or for bad. I think we have guns to protect us, to protect patients. I think in Europe, one strength we still have and we should maintain is a high level of academia, high level academicians, and still a very diffuse sense of ethics and a sense of public health. Public health is still high in our mind. That's, I think, is a good shield that even with inertia for a while will remain. If we also take action, probably on that domain, we will have a lot of more chances than others.
I don't think I can add anything new. I think that we have to keep the confidence that the society have put on us, being excellent on our work and responding to their needs. And the uptake of AI is just another tool. Experimentation is kept under control in sandboxes, but we don't uptake these new methodologies or tools without carefully assessing the benefit risk. That's our business, assessing benefit risk.
And we need to be careful communicating that this is what we do also when it comes to AI, I guess. I think that was covered in that question. So many of you, and especially you, Guido, in your presentation, stressed the importance of people and the staff at the agency. And there is actually an interesting question here. Maybe I can combine two of them. One of these, what are the biggest lessons from the past that you can relate to people working for the future? And the other one is new generations of staff are joining the agency and the network. Now I lost it. It's on screen. Good, yeah. What is your message to those joining the agency and the network, being exposed to this very complex and fascinating world for the first time. So lessons from the past, and how are we going to introduce the new generation to our fascinating world? Would you like to go first, Ema? You're looking at me like you want to go first.
No, I wanted to go second,
Okay,
actually.
so we are having Guido go first? Yeah.
Well, whatever I learned is useless now. But yeah, what I learned is that you have to involve people and to listen and to squeeze out ideas from them. Then you might play fair or unfair and say that the idea was yours, but the idea, you can't generate idea in isolation. So involvement of people, and sometimes the people hands-on in some process gives you more the key element for fixing things. I think this is the lesson I learned. And then having a consensus in the decision helps, but not go too much, because you will never have everybody convinced. And you have to take a decision at some time. And you do it wrong, so it's your fault. But you are paid for it.
Ema.
Yeah, well, I wanted to talk about, I wanted to give the message to new staff, new generations joining the agency and the network. And the answer is almost in the question. This is a complex and fascinating world. It is extremely exciting. I don't think people realize how exciting it is and what a role you can play. You can change people's lives. This is something we can do together. We can imagine the future. We can make it happen. And the only way we can do that is by joining together, but also by getting ideas from new generations of staff who have much different ideas to what I have. I'm back in the last century. I want to learn from new people, but I want you to know that this is exciting. This is somewhere where you can make a difference, and we do.
Anything you want to add to that from a network perspective, María?
I echo every word Emer said. I think this is a fascinating word. Where can you be more...
Full agreement across the
full
network
agreement.
here.
I only add a plea to younger generations. I know that we are going through very difficult times, that we feel threat for many different sides, that many young people feel they don't have the same opportunities that we had in the past. So they have probably a shorter view, and they don't engage for long term with anything, with jobs, with cities, and I would invite them to have a longer view. Yeah, because don't believe that we are so challenged, that we are going through the most difficult times. I could take here my parents, and you would be surprised of what are worst times. So please have a long view, look towards the future, and stay longer in your institutions, because that's the way to get the most of your colleagues and of your work and of your tax.
So, so before closing, I think I will give you the last word, Yann, as the patient representative. Any guidance, maybe for patient representatives, either joining the agency or joining the network as patient representatives? It looks like we in the future will have even more patient engagement in our decision-making process. So what's your guidance to those?
If it goes to the trilogue, as I say,
it
goes through. Yes, I think we've learned that to address a lot of the challenges, I mentioned thalidomide, but we could mention other parts of work. There is working groups on new methodologies, for instance, statistical methodologies and use of Bayesian methods. It sounds very dry, but in fact, getting the patient representative involved in understanding the science, understanding technology, understanding the methodologies is essential because they are the intermediate with part of the society. And that's part of building the trust. To address the challenges we've been saying, positive and negative, but this network of stakeholders that the EMA has developed, I think is fantastic. The network of experts, the network of patients' organization, of consumers, of healthcare professional associations. And we need to focus on that and allocate the proper resources because that's what we're going to need to address a lot of the confusion and on fake news and undermining of the trust into institutions and into the science. And we know that. But on the other hand, to speak to the young fellows joining the networks, I would really emphasize again what you just said, is that in my experience as a patient advocate, always look long-term, look 20 years. Sometimes people look at you like a fool, but that's the only way to think in our area, sorry, is 20 years, 10 years. But at the same time, always focus on making little or big things so that every week, every month, every year, you look back and you say, we did it. We made that progress, we obtained it. And then you feel empowered in moving towards your bigger vision. And that's what we need to get more of these colleagues on that page and to trust that making a change is possible and that there is no fate in accepting all the sadness and difficulties in the geopolitical, particularly situation we are, but that we're part of the change that we want to see.
Many thanks. Well said. So by this, I will thank you, María, Emma, Yann, Guido, for being part of this panel discussion. I excuse for having postponed, not taken 15 minutes of your lunch break. Lunch is upstairs, second floor in our restaurant, and I would very much like to see you back here 2:15. Enjoy your lunch.
You happy?
Yeah,
Was
happy,
was okay?
Yeah, more than okay. So, colleagues and friends, welcome back. I can hear on the buzzing that this was really a networking lunch. I hope you had lunch as well, but I definitely also hope you took the opportunity to network and meet with old colleagues, new colleagues, making friends and increasing your network. So this afternoon is bits and pieces of science that is ongoing, maybe science of tomorrow, preparedness, women's health, animal alternatives and so forth. But the first speaker we already seen once today, although wearing another hat, but this time it is the real Sandra Gallina speaking on her own, talking about how do we embrace One Health. And I'm sure you're going to explain for the very, very few, if there should be anyone in here who don't know what One Health is. The floor is yours, Sandra, please.
No, I don't think I will need to explain One Health, although we may have some discussions about definition of One Health, which is not as simple as it looks. In my usual self, I cannot read speeches. You must have noticed that too. I'm too short-sighted. So I will go with the first problem. The first problem is that we are interdependent. This is socially true, as I would say Guido was making the point in his speech. The social networks sometimes do cover up failings in terms of hierarchical structures, but you know, it's really true that humans and all beings are in the same broth. And you know, I was appalled to hear in Rome at a big do where we were discussing One Health, somebody who is an authority, I will not quote him, but the people who may have been in Rome may have noticed that I immediately said, you're wrong in saying us humans and animals. We are all in the same thing. Not too Darwinianly, but really because, you know, the environment puts us there. The paradigm shift has not happened though yet. You need to speak with your families and you will immediately realize that this thing of One Health is quite difficult to seize. They're not necessarily too convinced that what you're doing is really going to matter, till they discover that they have some antibiotic resistance, and you try to explain to them, well, perhaps you had too much chicken. I'm joking, but let me say, one of the experts from the, the SAM, we call it with this horrible acronym, the SAM. It's the experts that work scientific opinions for the Commission on very difficult topics. They did one big piece of, I would say, recommendations on, I would say, One Health. And one of the first topics is the whole of the antimicrobial resistance. And one of these experts had real antimicrobial resistance, and she almost died because they could not find an antibiotic that was working. It was barely the case that something in the reserve antibiotics worked. So she was really personally very much into this. But let me say to you, it's quite difficult. I would like to say that I am always objecting to people, including in the speeches of the Commissioner or in the speeches of anybody else, this figure of 35,000 people die every year out of it, because this is an estimate or a guesstimate. And in reality, what I have discussed sometimes with real scientists is that we may have much more than that. We may not be able to really put a real figure on the antimicrobial resistance, because the comorbidity of some elderly people that enter hospitals, wrongly so, because they are somehow parked there during summer and all, and get infections and can't be treated, it's never put down to antimicrobial resistance, but may be there. I'm just quoting again one example, but there is a whole host of, it's like the tip of the iceberg. So you hear this 35,000 and you say, okay, fine. Then you hear another figure which approximates more reality, which is the cost of antimicrobial resistance. This is a bit more sort of not scientific, but the accountants, the bean accountants know if you are absent from work or if you have sickness benefits, and they say that at least we in Europe are paying 12 billion every year, which is a modicum, and I'm somehow attracted to this number as closer to reality than the other one. In my view, when we work with OECD, I am trying to get, and I would like to thank them, Scarpetta, Colombo, and all of them, I'm trying to get many of these figures out of the cupboard of, I need a figure for a speech. No, we're trying to really check on them. And some of these figures may be very useful for me, especially for the discussions on money, on the MFF, because I am saying constantly I'm one of the few DGs that still sits at the table for the discussions of the budget, meaning I'm not a budgetary DG, but I'm sitting there because prevention, for instance, we are very keen on prevention and preparedness, and I have somehow approximately something in the order of 40 billion in terms of, so I'm beyond the trivial. I'm saying to people, if we could do prevention, we could be sparing quite a lot, and this is the bigger pot. This is not just, I would say, for the antimicrobial resistance. You must know that the Swedish presidency was quite instrumental in getting a beautiful recommendation out. I was honored to meet, I would say, the big professor on this issue. But he was actually a bit depressed because it was the third Swedish presidency, and he was telling me, we had so many presidencies, and it's the third time that we go for yet another recommendation. Sweden is, I would say, the best pupil in Europe, and frankly speaking, we are stepping up our limitations for antimicrobials. We are now going into the real world by saying a reduction of 20% of consumption. You know that there is a real difficulty in terms of getting consumption down or getting only sales down. And this is the whole difference. If you go for the sales is one thing, if you go for consumption. So 20% in consumption is a real target. Now, will the member states be doing the right thing? I tell you, no. If you read the latest figures, there's an increase in antimicrobials. I must say, after COVID is a disaster. So it's a plea to you all, please make people aware that by using antimicrobials, by using an antibiotic for a virus, I mean, they are really killing us all. This is not good. So I must say that the monitoring also pertains to EMA, so thank you for this, because we can keep an eye on the use and how it is used, but at the end of the day, we also will need to have new antimicrobials. And here we have a huge discussion in the Pharma Review. You may be aware of this, about the transferable vouchers, the famous voucher. I am one who says that if a Nobel Prize finds that the voucher is good, I don't object to it. I got a lot of flack for this, but I must say that nobody has come with better propositions. There is other systems, but I am hearing that also the subscription system has its limitations because it's extremely expensive. In our proposal, the voucher is extremely limited. We said that we can have maximum 5 vouchers for 15 years, and I tell you, I wish I could give the 5 vouchers in the first 5 years. This will not happen because finding a new antimicrobials is not easy. So again, the pharmaceutical industry will be a real, I would say, catalyzer of this whole discussion, because at the end of the day, innovation will need to be paid, and I hope we will find, if vouchers are not the thing that people want, then find another solution, but otherwise we will not have innovation. I'm clear. I would say that EMA has been, as has been recognized this morning, instrumental in, I would say, the vaccine big campaign. For COVID, it's still vaccine is an important word, because I may say that some of the diseases that we have to deal with because of the zoonosis coming over or for other infections could be stopped thanks to a vaccine. And here I come to the whole discussion that fascinates me with the scientists when we're trying to drive these very silly divisions between, I would say, the infectious diseases and the diseases that are not contagious and so on and so forth. And in reality, some of the conditions that become permanent conditions start with an infection. We have hepatitis that, you know, sprinkles from viruses, and then you may be getting into cancer. So let me say to you, many of these divisions are being blurred. What is important is that we go for, I would say, what we can use, and it's very important that we bear in mind that avian flu is something simmering next to us, and every day when I read news about the way that, you know, the defenses may not be working, including for COVID, I get worried. So we need to be prepared. We need to be looking at the possibility of having the vaccines that we need. I think it's important that we mention climate. Climate has received lots of resolutions, and it's moving the line of certain diseases upwards. Certain things that were not seen in temperate climates are now quite easily detected. There is a project in Italy that was quite good on the West Nile fever, and I must say, I quote it because, you know, this was the result of work by very different regions, and here again I would say, going beyond the anecdotal, going beyond the local, whenever Europe tries to unite at big, small level, whichever level it is, you are much more powerful, because with a hackneyed word I would say, diseases know no borders. You will not be safe just because you're a different country or a different region. Now, we have, I would say, quite some experience, because you also need to realize that my Director General, that has all the health that is above the soil, meaning we are in charge of health for humans, but also of animals, plants, and I would say the wastewater and all of that you may be discussing. We have had, I would say, very strange experiences, because I also have the largest auditors body. We go and audit in different things. We may be auditing zoonosis, but we are also auditing for antimicrobials. And I will say to you, the situation in Europe is very different. In some countries, where, you know, the levels are not particularly high in terms of revenue for the farmers. It's quite difficult to go and explain what it means not to use certain antimicrobials. They depend from the chicken that they raise, and they say to you, frankly speaking, we can't go without this thing. So we see the things. We are not completely, I would say, blind. We have fact-finding visits also around avian flu. And with my services, we will need to do a plan, because, you know, one of the first things that the farmers will need to know is what to do if there is a cow that produces milk, and where does that milk go if the cow has avian flu. In Canada, they did quite a good thing. We have had a huge, a series of meetings which I call jumbo, with vets and, I would say, public health authorities. I consider that one success of my career, because it was difficult that the two worlds speak together, and still they need each other in this one health perspective. And one of the first problems is that you need to reach the farmers with public health ideas. We had this experience during COVID with the mink, and praise goes to Denmark that did the right thing, culling all the mink at the moment when there was this tragedy of COVID. The Prime Minister didn't hesitate one moment to go for that very harsh choice. So let me say to you, you need to be aware that One Health is permeating you more than you think, and in different ways. There are two things that I want to mention. One is that I immediately decided that I want to have one specific, I would say, part of my directorate that will be called One Health, because, you know, we need to be the pole of conjunction for the whole of the Commission on One Health, which is not an easy topic. People don't see the links, but then when you start working together, you at least start to build relationships. And we also had this fantastic idea to put 4 or 5 of our agencies together, including EMA. We have Frank here from EFSA, he is there. We have also, I would say, ECDC, which is an important agency in terms of public health. And I would say we have also the environmental, I would say, agency that was also one of difficult for them to work together at the beginning. Now it's getting better. So I'm being very honest with you. Working in silos is very practical, it's very easy, but working across the silos is what is very difficult. And I would like to say this is why One Health becomes a difficult topic, because you need to stretch yourself a bit. So thank you to EMA for stretching a bit beyond EMA and to do the work with us on this difficult topic. Thank you. Thank you.
Many thanks, Sandra, for this You don't need to take any questions. No, for sure. You answered all the questions. I decided. That's the privilege of the chairman. And thank you for being here, because I know you will have to leave us before the end of today. So the next presenter is a rare species. First of all, he's a nice Swede. And that is only something a Dane can joke about.
Yes.
Maybe the Finns can do so. We can do that afterwards. The other thing is that Mats Velin, who is a Senior Director of Biotechnology at the Swedish product agency, the MPA, attended the first meeting for biotechnology products back in 1995. And Mats is still a member of that working party. So here we have one who can explain what has happened in biotechnology for the past 30 years, and maybe look a little bit into the future as well, Mats. The floor is yours.
Thank you. Oh, great. Yes, to start, I'm very honored to be invited to come here to try to give you some ideas of what has been discussed within the buildings you see on the right side over the last 30 years. So I start with this one, which is, title is Ad Hoc CPMP Biotechnology Working Party EMA, Westbury Circus, April 95. This was the first meeting of the BWP in London. I think someone has told me that that was also the very first meeting in a working party or committee at these new premises. And as far as I remember, I had the same tie. But that is more or less the only thing that hasn't changed. Or this hasn't changed. I don't wear this each and every day, but it's the same tie. But if we're looking at just this one, it's no longer an ad hoc group. It's no longer a CHMP group. It's a CPMP group. It's a CHMP. It's no longer biotechnology. It's biologicals, and it's no longer EMA. EMEA, it's EMA, and it's no longer Westbury Circus. So things has happened, but it's not only there, because when we see what we did discuss at that time, the BWP at that time had started in late 80s in Brussels, and there was a lot of discussion on viral safety of, in particular, plasma-derived products. So we had, in this very first meeting, there was long discussion on harmonization of blood donor screening, nuclear amplification techniques testing, virus validation. We had Creutzfeldt-Jakob's disease. There was some few hours of breakouts for discussions on dossiers. This is quite different today. Today is I would say 80% or so, maybe even more, is discussing on central scientific advice and new applications. And the time put to discuss the science is less at the meetings, but that is then handled by drafting groups where people who are really involved, which may also be more effective way of doing it, in the sense that the people that really know, and then it's told to everyone. So that is, so if we just go a little bit over what has happened then. So if we go even before 95, that you had the proteins, some of them came from body fluids or organs. You had insulin, you have human growth hormone, and then you were able to do it by recombinant technology. The first ones came in the early 80s, but we had the process defines the product, which had an impact on the requirements to do some changes to the production, because then, but you can't really show us that it's the same, so you may need to do clinical studies with your changes, which of course then will sort of lower the interest of doing the changes. But then we had you have an introduction of comparability for quality level through an analysis of physicochemical properties and biological activities, and showing that maybe you don't need clinical studies. We have an EU guideline in 2003. We have ICH Q5E in 2005. And then, then we had the first approval of the biosimilars coming into that. Yes, not only you do a change of your own product, you can compare to an originator, showing that this is considered to be the same, and with limited clinical trials, you can get an approval. And the first ones we had was Omnitrop, was a human growth hormone. We had non-glycosylating, quite short proteins, like less than 200 amino acids. Now we have much more complex proteins, and I'll show you a figure on the monoclonal antibodies that they are 1,400 amino acids. They are glycosylated and much more complex than we had. And what we have now is the discussion on the need for clinical data for biosimilars, depending on the and then I realized that this is not the last one of my presentation. Sorry, but I think it was no. So there were no need for clinical data without sort of tailored PK pharmacokinetic studies to be really correct here, and we do see some synthetic versions of peptides earlier produced by RDNA, and many of those that was discussed earlier this morning with 30, 40 amino acids are now possible to do by the synthetic routes. So if we have some milestones which we have gone through, that before EMA we had the classical CGD from human growth hormone. When during the sort of late 90s, you had new variant CGD, which was linked to the BSE outbreak due to changes in treatment of bovine material. That has a huge impact on medicinal products, that all products needed to be certified to be safe from a TSE perspective. And that, you could imagine that all of them, it was a huge work, and it was very well supported also by the European the Pharmacopoeia having their certification scheme that meant that companies could come there and then they had a certificate of their particular constituents, and that could be used for everyone that used that one. So it was really a useful one. Again, I've already mentioned biosimilars, which is quite a different change from where we were, and that is, I think it was triggered by Omnitrope, because that was initially applied as a well-established use, and then there was the discussion, is this a doable way? And in the end, it was a no, it's not, but then the biosimilar or the similar biological substance, I think, or product, was introduced. And then one very important one is introduction of ICH Q8 to Q11. That is late 2000. That is a lot of instead of testing in quality, you have quality by design. You have criticality ranking and other things that really go for what matters, and that has really changed the way of testing and looking at things and say, okay, you may not need to test because you have shown that this is of no really importance, and having that together with, I would say, ICH as a whole has really been very important, that we are working and companies are working to different regions and coming up with a guideline that will be more or less acceptable by everyone is a great benefit and a good work. It takes a lot of work. Ask me or ask Jean-Louis, but it's and I think also ask Emer because you were involved earlier as well. But then we had 2009. We had the influenza pandemic. Everyone thought that this will be avian flu, H5N1, because that was what we have seen in China, and people were dying, but not that many. Then so the guidelines were prepared. There was an administrative aspect, how to do it, that you submit sort of an core dossier or whatever. This is the way we are going to do it. But then when the actual strain comes, we are going to introduce that. So in that sense, we were well prepared. But then when it came, it wasn't an H5N1, which I think was a good thing, in the sense that wasn't a killer that we would earlier expected it to be. So there was also, but in particular Sweden and others, there has been a handling of side effects post-pandemic on this one. Then we had a lot of very good meetings and workshops with industry and other agencies. And I think particularly like 2017, we have had a use of prior knowledge. And 2018, there was one on accelerated access that for unmet clinical need within sort of prime and breakthrough therapy in US. And the latter resulted in a joint guidance from us, which I think is really, really useful for companies in the sense that, yes, we can play the same game to both, and they are quite, and yeah, we have with the Brexit, as was mentioned by Guido, and the movement of London to Amsterdam, of course, had some impact on our work, concentrated more or less totally on running the applications and so on for very good reasons. And then 2 months, I think it was after coming to this building, you had the COVID. And you have really to change the work. You couldn't come here. You had to work in a completely different way. We saw some new vaccine modalities, and we could really discuss acceleration there, because the efforts put down on approval of products is marvelous compared to what we have had before. And that is, then you could say, yes, these two workshops that use of prior knowledge, and also that what could be acceptable in an accelerated situation where you don't have anything for that clinical need. So we were actually quite well prepared for that. So just going through some of the types. So the first monoclonal was approved in Europe in the late 80s. It was produced in mice ascites. It appeared quite, with the methods at that time, it appeared quite pure. It wasn't. The second one came in 94. There were 2 more in 98. 2005, we have 16 monoclonals. 2015, we have 52 and 1 biosimilar. And 2025, we have 210 monoclonals approved in total, of which 61 are biosimilars. And 12 of them are then also antibody drug conjugates, and where you link a chemical drug to a monoclonal to really have a very high specific therapy. And why is that? I think initially that, yes, you can do it and you can direct it somewhere, but you didn't really know how to make a therapeutic use of it. It was used to some extent for diagnostic purposes with some radio labeling, but then more understanding of what can they do, and also we had improved analytical methods, which gave some meaningful comparison between the biosimilar and the originator, and that is why we are seeing this huge number of new biosimilars coming up. For vaccines, which is close to me, I think this is by far the biological medicinal product that is given to most people. And it's different in many other products, because this is given to healthy people. This is very often given to children and healthy children. And safety aspects is therefore very primary here. And if we would have difficulties there and the trust in vaccine would be poorer, then we would be out on thin ice, I think. So it's interesting, we have a very long history from the late 700s, but what we have now, we have combination vaccine where you can combine antigens from multiple infectious diseases like diphtheria, tetanus, pertussis, polio, hep, or you have antigens for different serotypes of the same bacteria. And we have, I think for the moment, we have 21 valent pneumococcal polysaccharide conjugate vaccines for the time being. We started with 7. We have virus-like particles for the papilloma vaccines. We have the mRNA vaccines, and for example, then 2 of the COVID vaccines were there. And then we also had the adenovirus modified to contain a gene to making a specific protein. So it happens a lot. We are working a lot with new adjuvants and so on. So this is a very interesting field, I would say, which is also the same thing for cell and gene therapy. That, yeah, I'm not going into the background, what they are, but what we see is that a lot of things now come, which is a combination where you put in the sequence you want into a cell, and then it can be express a new gene, so and then given back. So this is really a game changer. That all of a sudden it's possible to treat what in the past used to be untreatable diseases. And I had a look at what is then for Prime. Currently there's total 79 products. ATMPs is 35 of these. Biotech, other biologicals are 25, chemicals are 19. It's really that with this type of products, we can really medicate a number of diseases we couldn't some years ago. And we see a similar scenario for the approval as for the monoclonals. The first one was approved in 2009, next one in 2013, 2 more in 2015, and now we have another 19. So it happens a lot. And what is then to be expected in the future? I think a lot. We will see more of the ATMPs for good reasons. We would see personalized medicines, including 3D printing. We will see the antibody drug conjugates even more. We will have point of care, bedside manufacturing, where the final part of the preparations will be done close to the patient, and that will be in particular ATMPs. We are making use of models, digital twins, in control and development of new products and their processes. And this may speed up and lower costs for development of new product and improvement on old one. And we also see, as we said, I think it's clear we will expect more mRNA products as we have until now. So having said that, ATMPs and antibody drug conjugates and some of the others are quite expensive drugs. And that is, so the case is, yes, there is something, but is it possible? Is it at all possible? Will this be used? Because the treatment per year is really high, but let's see what can be done. At least it's there. We'll see what will come. And then, of course, there's a lot more, but we don't know yet. It may be that Marco will have some idea in his presentation, but I think, or I know that this was the last. So thank you very much.
Thank you so much, Matt. Would you be okay answering some questions if there would be someone in the audience here? While audience think, I will ask you one. You raised the issue about affordability of ATMPs,
Yeah.
But you also mentioned biosimilars. Will we have biosimilars to ATMPs?
That's very good. question. I guess the biosimilar, in the sense that biosimilars themselves had evolved, that earlier on you could do 150, 170 amino acids shown this, and non-glycosylation. Now we do it for there. We need to have a better understanding what exactly in these 18Ps are the important one, and how can we do it. I think we have a number of them which are quite comparable, but they stand on their own legs for the time being. So maybe in the future, but it is a tricky question, because it's also that companies are putting a lot of money in it, and some they are not coming to it because they are stopped in phase 2 or 3 or so. So it's a lot of money put into it, and how can we do it? So it is something we need to consider, because I know that we have said, okay, this would be perfect for this patient, but the regions cannot afford it. So that's a really issue.
Any question in the room for Mats? Otherwise, we will thank you so much, Mats.
Thank you.
Thank you. So our next speaker is Thomas Hartung, who's a professor at Johns Hopkins Bloomberg School of Public Health. And he's going to speak about alternatives to animal testing, and also a bit of AI. And actually, we talked yesterday, because when you submitted your presentation, some EMA colleagues have concerns about the pictures and the figures in your presentation, whether they were copyrighted or not, because if we're going to publish this, and you told me last night, there's no copyright on any of your pictures because they are all AI generated.
Yeah, that's the modern times. So first of all, happy birthday. I had the privilege to work with EMA since 2002, when I joined the European Commission to become head of ECVAM, the European Center for the Validation of Alternative Methods, so exactly the topic I'm still working on. And EMA has been a trusted partner since, and I really appreciate to have the opportunity to be here. The topic is actually much more AI, not just as an afterthought, and if I had given the title myself, I would probably have said, AI will only replace regulators who don't use it. So, I made my slides available. The QR code is also on the last slide again. I'm directing since 16.5 years now the Center for Alternatives to Animal Testing at Johns Hopkins University, so I changed the side of the pond. But I also co-direct the Center for Alternatives to Animal Testing in Europe at the University of Constance with Marcel Leist. And I'm also field chief editor of Frontiers in AI. My quest is to replace the dance around the golden rat and retire these type of animal models. When I joined the European Commission, I got the job because I had just successfully validated what is now known as monocyte activation tests and had developed one of these assays myself. These assays are now replacing in Europe alone 150,000 rabbits per year, but it took us almost 30 years to get there. Yeah. So this is showing you something. There's potential of modern technologies, but it is slow. And I want to show you that these things are accelerating because we have new technologies. Also, as a pharmacologist and toxicologist, did my share of animal experiments. I never liked them, but I did. When I was charged with replacing animal testing, this was mainly because of ethical concerns. Yeah? Find something which is the same or hopefully better, but mainly ethically. There's people who want this to be replaced. But the longer I'm in this business, the more I see this as an advance of science and an economical question. Yeah? These tests do no longer serve us well. This is an article I wrote last year, targeting a general audience about the misleading role of animal models in drug development, where I show that from a lot of studies combined, we have to assume no animal test is more than 80% accurate. Many of them are much, much less so if you compare the same experiment done on mice and rats, or rats and rabbits. It is typically between 60 and 80%. And if you use such tools to find a drug to go into human trials, it is very often misleading. And this is alone should open up for other tools. The other thing is I never liked this war of faith, that there is some people who accept animal tests and defend them vigorously, and other people say we have to get rid of them tomorrow. So I thought early on in 2002 already, can't we do something similar to clinical medicine, where evidence-based medicine has helped us to be simply factful, demonstrate what is the fair place for each technology and compare them. So we created the evidence-based toxicology collaboration. In 2007, still at the Commission, we organized the first international conference and we got it started. Since 2009, I have a chair for evidence-based toxicology in Johns Hopkins. And the collaboration we created 2 years later enjoys strong support from agencies like the FDA, the EPA, but also our president is from the European Food Safety Authority. And I would very much like that EMA is joining the club in our board of trustees and others, because I think it is a timely thing to do to handle more objectively and transparently whatever facts we have to handle. So, invitation, number one. To just show you with one slide the difficulties and some of the problems, the chemophobia we have been produced with some of these animal tests, I give you the example of the cancer bioassay. The cancer bioassay is still the only way accepted to show whether a substance is producing cancer or not, and it is hardly done. Now, we have about 3,500 of probably 350,000 marketed chemicals which have ever done a cancer bioassay. And the reason is simply, it takes you 4 to 5 years to get the results. You pay about $1 million. You need 400 animals, and you need 10 kilograms of the substance to treat these animals for 2 years of their lifetime. Tell a chemist to produce 10 kilograms of a substance which is not yet in production. And the results you get are terrible. 53% positives. We estimate that maximum 5 to 10% of these substances are actually carcinogenic. The reproducibility between mice and rats is 57%, which is not really very much encouraging that they predict humans any better. And not even the organs in these animals are anyway correlated, the one in the kidney, the other one in the liver, and so on. So it is not really a value proposition to use this assay, and I'm so excited that ICH is finally tackling this to make this something of the history. In general, it is all about now transitioning to new technologies. This is an article from last month where I wrote about how this is similar to 110 years ago when horse coaches were being replaced by cars. And the type of arguments you hear, how reliable the horse is, how much infrastructure we are missing for the new technologies. And I very much hope that in a few decades, people will smile about these antiquated arguments and say, how could they ask about not using AI, not using modern technologies? So, from my point of view, there's at the moment a number of disruptive technologies. They're disruptive because they're upending the way we are doing things so far. And I will focus only on 2 of them. One is the microphysiological systems, the organoid and organ-on-chip systems, mainly fueled from stem cell technologies, and then AI and data analysis technologies. Both of them feed into AI in making sense of these big data. So we have, on the one hand, the possibility to produce human-relevant data on scale, and at this other time, we can analyze now data, because since 2 years, actually a machine is better capable of reading a scientific paper and annotating it than a human. But it's not limited, like my PhD student, to 1 or 2 papers per week. It is millions in a day. And these systems do not forget. And this is a tremendous value proposition. And this is allowing us, first of all, something which in the chemistry field we call green toxicology. In medicines we call this fail early, fail cheap, because we can use these technologies not after we spend almost $1 billion for preclinical development, and when we want to go into humans, when we do our 10 million package of safety testing, but we can do this even before we synthesize a substance if we have reliable computational methodologies, and we can do it on micrograms of quantities we have in our hands with the MPS type of system, the microphysiological systems as the EPA is calling it. So these technologies are allowing us to front load already these concerns. The second side, which I think is really getting more and more interesting, and I will come back to both of them, is that we can combine this also with exposure sciences. Our mass spec systems are getting about 1 log order more sensitive every decade. We're finding more and more substances in the biospecimens, in the blood, in the urine, in biomonitoring studies. And again, it is the AI, the exposome intelligence, which is helping us to make sense of this and can lead us to human exposome or to personalized medicine, where we consider a treatment not as something for everyone, but something for an individual with a specific disease history and its unique aspects, which we more and more integrate by digital twins. That's the big, let's say, 10,000-foot perspective. Let's talk about how this vision for safer products, better treatments, and safer people can actually be implemented, and how these technologies are at this moment lending themselves already to do so. So the first technology, which developed really over the last 2 decades, is microphysiological systems. That's the umbrella term coined by the FDA for 3D cell culture systems, organoids, organs on chip systems, multi-organ chip systems, patient on chips, and others. My group has been accompanying this over the years. We started with the first workshop report in 2014. We had several MPS workshops dedicated to the future, bringing opinion leaders worldwide together. The last one published just in April. We developed good cell culture practices for these systems, so to have quality assurance, and we started a series of world summits in 2022 in New Orleans, 23 in Berlin, 24 in Seattle, and just 2 weeks ago in Brussels with 1,500 people, a very successful meeting. And you can see here how the number of attendees has reached almost 1,500 now. The number of exhibitors is steadily increasing, a very vibrant activity which we had in Brussels, and we're looking forward to go to Washington in May next year. This is me handing over the baton to my wife, Lena Smirnova, as the president of the society who took over the organization of these things. And so my team was last time responsible for the meeting. So our interest into organoid systems came from this steady increase in autism in the US. You see here the latest numbers from April this year. Now 1 in 31 children in the US is diagnosed before their 8th anniversary with autism. 1 in 31. This was 1 in 10,000 in the 70s and has tripled over the last 20 years, where no change in diagnosis also took place anymore. And this cannot be genetics, though there is a genetic component, there must be more of this. So, and it seems even to accelerate if you see the latest numbers, which might be possibly even a consequence of the pandemic. So we already 20 years ago started to work together, and as I say we, my predecessor in CAT, Alan Goldberg, and me at the time in ECVAM, and with 5 international conferences and 9 workshops, we addressed this problem, because there is only an animal test for developmental neurotoxicity, which takes you 2 years, but more than 1,000 animals, and thus cost 1.4 million to carry out on a single substance. Not really a value proposition to find and screen chemicals and drugs for their contribution. This is a paper from last year where we describe these 20 years, where really a group formed and ultimately led to OECD guidance on how to use in vitro systems for this purpose, because really a community out of nothing was shaped to address the right systems. And this was the meeting our European branch carried out last April in Constance. We developed out of this the interest into brain organoids from stem cells. We were not the first to produce them. We were beaten by half a year, but we were the first to mass produce brain organoids. So we really get about 4,000 human brain organoids in a 6-well plate. Yeah? That's about a cent of one of these. Yeah? And most experiments can be done on a single brain organoid. And these systems are not only having all types of neurons we have looked for so far, which are electrophysiologically active. We have oligodendrocytes myelinating the axons, a very rare thing, a very important endpoint. And we have astrocytes, and with the latest protocols, we get the glial cell population to about 50%, which is physiological. And all of them together, standardized, you see the size distribution, Here, the reproducibility in cell populations we must produce, and we can do this of different patient backgrounds. So, for example, we have 20 lines from autistic children, and they are matched controls we are working with. We can do with these things which were not possible with animals. We use this, for example, to show first already in May of 2020 that COVID-19 is infecting human brain cells, because we had the system and our colleagues had the virus, and it was easy to check. But we also showed, for example, in this example, a paper from 2021 that a risk gene for autism, SHAD8, and chlorpyrifos, a pesticide which is known to have some liabilities here, synergize in producing a phenotype which we would call autistic in our brain organoids. And this then led the NIH to create an Autism Excellence Center. We are now monitoring 175,000 children in 18 centers, and they form exposure gene hypothesis, and we are doing this then in our brain organoids to verify experimentally, is there really such a synergy? Something you cannot do with any animal. And this is perhaps the most science fiction type of research we're doing in this field, because we are combining our MPS via electrodes, microelectrode arrays, with an AI system. This little jumper here is actually controlled by a brain organoid. And it is learning to jump at the right moment to avoid these obstacles. We use this video environment to illustrate what we're doing here. These are only partially published results so far, but we have received about $2 million from Hopkins already to invest into this with our partners, and there's an increasing batch of articles around this system and how we're making progress in developing memory and cognition as a very simple form of cognition. You can imagine what this means for pharmacology and toxicology. We call it organoid intelligence because this is possibly giving us a system of memory and learning. Oops, I have to get back, sorry. A system of memory and learning, which we can do with patient backgrounds. We're doing this with Alzheimer patients, brain organoids, and others, and we're looking into, do they learn differently? Do they get better? Do they memorize it until for the next day that they improved the game? I think this is showing you quite nicely, and this is bridging for me to AI. This is showing you how human skill levels have been achieved in different aspects of human capabilities. The baseline is the AI system is worse than any human, and the 100% line means the AI is outperforming any human being. And you can see to the very left of this picture here that the, it's working actually. This is human handwriting, deciphering human handwriting. The AI took 18 years to learn this. But on the very right, you see in orange, for example, coding. In only 2 years, AI learned to code, to program computers so well that last year, about 80% of all coding was done by machines, and all of the big tech companies have laid off coders. Yeah? In only 2 years, they outperformed humans. And it's only a matter of time for me until toxicology becomes also an AI-driven technology. And pharmacology actually was already a bit earlier, because since 2019, roughly, AI was good enough to design some drugs. And in 2023 already, 18 AI first drugs, so AI suggested drugs made into human trials, phase 1. And last year, the first one successfully passed a phase 2A and went into phase 2B with extended patient numbers from in silico medicine, a lung fibrosis medication. And imagine this, since 2019, that's when their project started, to only 2024 for preclinical development in such complex disease. This shows you the value proposition of these type of tools. 1 day earlier to the market is worth about $1 million. Yeah? That's what it is. So I've written extensively about the possibilities of AI in toxicology, how toxicology became a big data science with the high content omics and imaging technologies, high throughput robotized testing, sensor technologies, literature, and so on. And in the end, I think that we will soon have to write toxicology with AI. And the quick definition of this is, for me, toxicology is making big sense from big data with big computers. And that's what is at the moment entering. And alone the data retrieval aspect of this is incredibly valuable. I was peer reviewer for the EPA of a risk assessment on a Superfund site chemical. 2 people had worked on this for 18 months to bring together all of the data on these data poor substances. 2 people, 18 months. After I did my peer review, for the fun of it, I used the AI systems and I found all of the sources in 1 hour. And I found even a complete risk assessment the EPA colleagues had missed, done by the fragrance industry. So you can see the value proposition of just getting the data and being more comprehensive. But then it can integrate pathology data, multimodal things, and help us with our reporting. To give you a bit of flavor, BioBricks is a project of ours where we have this paper, I think is being hopefully accepted these days, is a way of importing entire databases with 1-line command. And we have written codes and shared already mostly on GitHub that with 1 line you can import an entire database without knowing how it is programmed, and you get in a standardized way all of the information. We have 140 of these. The first 12 of these databases gave us in total 117 million chemicals for which we have a chemical, a property, and a result. And 254 million of these triplets were generated on these 117 million data points. That's a treasure trove. 4,000 of these activities have more than 1,000 entries, which means for us it is good enough to cover the chemical universe. And these 4,000 properties you get in seconds, and the average accuracy is 91% in the training set. Compare this to 60 to 80% accuracy for 10 animal tests. So it is really about, at the moment, about building trust into these systems and making sense of this abundance of information. Suddenly knowing 4,000 things about a chemical is not making your life necessarily easy, because you need to interpret these 4,000. This is a paper with Rob Califf, the former head of FDA, which came out just in April, where we're discussing how regulatory science at the moment has to live and stand up to this artificial intelligence. And this is also a series of articles about the aspects of validation, the challenge of these new methods to validate them. It is showing us how AI can help us, but also discusses how we can possibly validate AI systems with their enormous change. One article is discussing how biomarker qualification, something close to your hearts, is possibly helping us to make things more human relevant. And I call this altogether next generation validation for next generation risk assessment. Important discussion. Let me quickly close with the 2 aspects I addressed. Green toxicology, so benign by design. Alex Mertens in my team is leading this. She wrote the first textbook on green toxicology, is an assistant professor now. And only 4 weeks ago, we met in Stockholm for a Nobel Symposium with the fathers of green chemistry, Paul Anastas and John Warner, behind this. And we did the Stockholm Declaration on the Future of Chemistry. And this is all about using the same technology, as I said, front-loading them. And this is the same thing like what investigative toxicology is trying by prospectively risk anticipation of the targets and chemical classes which are being used. This is a workshop we did with the pharmaceutical industry. And I see that especially now with more than 50% of the drugs being human antibodies or human proteins, it is of highest importance that we get microphysiological systems and others into use in order to carry out this work. The last aspect for me is the big integration. I believe that ultimately we have to integrate genetics and exposure. This is a very easy line. The genes load the gun, the exposure pulls the trigger. And by mapping the human exposome similar to the human genome as a counterpart, we could put a safety on this. And with this idea, we carried out in May an exposome moonshot forum in Washington with a lot of European colleagues, endorsed by WHO, UNESCO, the OECD. We had a lot of high-level speakers there. And in this meeting, I think we really set a starting point towards creating a human exposome project. Fernand, my deputy, is doing this. Again, AI is in the center of a lot of this, because only AI can help us to understand the complex biomonitoring data and the patterns we see. We did sign a declaration towards a global consortium for the human exposome. with the 300 participants we had, and I hope that this will be the starting point of something big and new. The last slide and statement here, I think that a lot of signs are on green. You might have followed that in April this year, the FDA announced a roadmap to reducing animal testing, singling out monoclonal antibodies as their first target, but giving deadlines for moving out of animal testing. The very same day, Seldin, the new head of EPA, renewed their pledge to move out of animal testing by 2035, something already done by Andrew Wheeler in 2019. And 10 days later, the NIH came with this press release on to prioritize human-based research technologies, the microphysiological systems, to go hand in hand with this. Europe is at the same time talking about a roadmap already for a number of years. We are expecting a roadmap to phase out animal testing by early next year. So I put you on yellow, not yet, yet quite green, but we also saw already that the European research area, which was just announced, has singled out this as one of the priorities for moving forward. With this, I hope I have shown you it is now or never. We have some disruptive technologies which we can integrate with existing knowledge by AI. I think this is a value proposition for accelerating product development and is going to optimize prevention and healthcare. I hope that the prospects of human exposome and a greener production, a production of substances which are safe and sustainable by design, as Europe is calling it, is a prospect which helped us all. Thank you very much, and I hope we have time for some questions.
Many thanks, Thomas, for a very inspiring talk here. I don't know whether we are on amber when it comes to moving out of animal testing, but we are close to getting green. I don't think we have made so much noise about it as other parts of the world have done. I'm pretty sure we have a question here for you because some of our audience was attracted by your brain organoids. And when is an organoid suddenly being a real brain?
I think that cognition is a functionality of the brain. And the big thing about microphysiological systems is to recreate architecture and functionality. But the fact that these systems can react and learn something does not yet mean consciousness, sentience, self-awareness. We are very far from this. But we are working very closely with a number of bioethicists. There's an increasing interest on their side, even going into experimental ethics to see how much it takes to build a system. Our standard brain organoids are 500 micrometer, have 50,000 neurons, which is about as much as a housefly. And nobody will believe that they are being trained only on some very simple computer games could have any way of consciousness or self-awareness, but we have to assume that this can happen. Our largest systems we are building at the moment are 1 centimeter, and this is already 2 mouse brains, and they could very well, if we feed them with more information at some point, assume some type of consciousness. Yeah.
Interesting, definitely. Other questions for Thomas? There's one coming up here, yeah? Number, these are linked to climate change by using...
Yes, the exposome is not meant to be just your chemical environment. It is also the social environment. It is the physical stressors. So there is subgroups. There's a lot of climate change exposome work. We also have discussions whether it's only about humans or whether they have an ecosystem, which is also to be integrated. It is clearly starting with the human exposome to match the human genome at the moment and also to integrate here, but we should be open for all of this. There's also increasing interest in microphysiological systems from animal species, from fishes and others to see whether we can create possibly information sources very similar to the human systems I'm working on.
Excellent. Many thanks, Thomas, and once again, thanks for coming and giving us presentation. Our next speaker is my colleague, Corinne de Vries. Corinne is head of our translational science and soon to be the EMA liaison at the US FDA. You're going to talk about gender equality, women's health, a forgotten priority. You and I have sometimes talked about we did all this legislation back in the 60s due to the thalidomide catastrophe. And the only subpopulation that haven't benefited from this is actually the females and the pregnant females. And those were the ones we wanted to, have you been overprotecting them? The floor is yours.
Thank you very much, Stefan. Okay, this is easy, I can do this. Thank you very much, Stefan, also for giving some airtime for this topic, which of course will be, what I will be presenting is results of work of 30 years at this agency, but also beyond and together with stakeholders. And actually not just the agency, the fact that this work has been possible is because of also the relentless support of committee members, rapporteurs, committee chairs. I'm looking at one particular. So it is a tremendous honor that I can be here and present some of this work. So gender equality goes beyond pregnancy, women's health. There is a lot of evidence out there that there is no gender equality. Women are, on average, they live 5 years longer than men. But the healthy lifetime that they have longer is only 4 months. And so there's work from OECD, Eurostat, World Economic Forum that shows that's because they have longer chronic diseases. There is a diagnostic and treatment delay, and that's because they have less money and they spend more time looking after the men in their lives. And so as an agency, we get questions about this. So is it true that for EMA, women are the forgotten priority? Oh. Sorry, there was a copyrighted slide in there, I think. For EMA, they can't be the forgotten priority because the clinical trials regulation dictates that the clinical trial population needs to represent the anticipated user population. And so women have to be included in clinical trials if they are going to be impacted by the disease. So what I'll start with is present you with some data on the inclusion of women in clinical trials. And I start with the first slide, which comes from data from EURORDIS-CT, which was a trials, that is a trials repository before CETIS, which was presented by María earlier. And what you see is, it's not an error here. 90% of trials include both men and women. 4.8% include women only, 5.2% include, 4.8 include men only, 5.2% include women only. And the tiny bar on the right, and then an invisible bar on the further right, is the pregnant and breastfeeding women in clinical trials. So that's the total, but does that mean if you have 90% of trials include men and women, does it mean that, okay, but these trials have 80% men and 20% women? That's not the case. So women are actually, overall, this is a slide, you can't see it, but what you see on the y-axis is the proportion of women represented in clinical trials per disease areas. And this is a study carried out in 2007 by colleagues from EMA and the network. Really demonstrated that there is overall appropriate representation of women and men in clinical trials with some slight differences. Overrepresentation of women on arthritis, rheumatoid arthritis, and on allergic conjunctivitis, slight underrepresentation of diabetes, hepatitis B, and hypertension. But when the authors then looked at sex-treatment interaction, it didn't actually result in clinically meaningful results to the extent that it required labeling or dosage changes, different dosages for men and women. That's the clinical trials. Then they also carried out a literature review, and we have done further analysis since this publication from 2007 to keep checking whether this is the representation of women is appropriate. And what this shows is that actually, yes, there is some biased inclusion to some extent, but most of it is not clinically meaningful. And when it is clinically meaningful, it does result in differences in labeling. And we're totally transparent about it. So you can find this information in the EPAR, you can find it in the SMPC. But that doesn't mean that there are no underrepresented population in clinical trials. And so there is an IHI project currently, has just recently started, that really looks into more overarching inclusion, fair inclusion, appropriate inclusion of all population in clinical trials, not just men and women, but different social and ethnic groups, and so on. So we're really looking forward to seeing how that work progresses. The other thing is that, and I'm so happy that Thomas gave his presentation, because the other thing is that what is also true, that is treatment responses and treatment pathways sometimes differ between men and women, and that's also the case in animals. And so it may be the case that if you start only on male rats and a pharmacological product is not effective, it never gets developed any further because it might be effective in women, but we wouldn't know. So there is some areas of question there, but yeah, that is something for us to bear in mind. Now the other thing is that, so the clinical trials information system really helps us because it'll enable, it gives us the opportunity to keep this kind of analysis going forward, but also it means that we're completely transparent about what clinical trials are ongoing throughout Europe, and it means that people have a searchable database that enables patient enrollment. So if a clinician or a patient wants to know, is there a trial currently ongoing that might benefit this particular patient, then there is a way of searching for that. And Guido, you mentioned transparency, and María, you mentioned ACT-EU. And with one of the priorities in the network, in the regulatory network strategy, being equitable access to medicines and also clinical trials. Now, ACT-EU, this accelerating clinical trials in Europe, benefits from multi-stakeholder interaction. We have 43 different groups of stakeholders who we really interrogate and ask what should be the priorities. And equitable access has been identified, but women as underrepresentation in clinical trial has not in any of the discussions been identified as an issue. Now, the nice thing about the CTIS system is that if people want access, like I said, you can find it, and you can even do it with the clinical trials map. So now I go towards what Stefan alluded to earlier. The big group that is missing in these clinical trials is pregnant and breastfeeding women. Now, why is that the case? And Stefan, you mentioned thalidomide. And I ask you to just ponder this for a minute. In a clinical trial, you give an experimental treatment to patients, you randomize the treatment, and then you evaluate the analysis, the results of this experiment after. And so the question I want to pose to you is it okay to experiment on pregnant women or on breastfeeding women? And generally, when you ask this question, the immediate response is, of course not. How dare you suggest it? But there's actually a delayed response that, well, actually, there may be occasions where this is appropriate, or it might be appropriate. And so there are 5 million babies born in Europe every year, and 90% of women use some form of medication during pregnancy, which has not been tested mostly in a formal clinical trial setting. Now, that includes things like antacids, folic acid, and so on. But 25% of women use a medicine for chronic diseases, such as epilepsy, asthma, diabetes, and so on. And so what we're doing in the agency, so when we say we don't want to experiment on pregnant women, here are some of the examples that have really shaped our thinking. There are products that have been developed, given for indications, considered where the product is considered necessary, and now we're scared because some of those have really had detrimental impact on the baby. So we go, oh no, we don't want to experiment because you don't know yet what's going to happen. But when you do that, when you think like that, you're actually blinding yourself to the fact that there are diseases that are also detrimental during pregnancy, that have a really detrimental impact on pregnancy if not treated. So for example, diabetes, if left untreated, gives bad impact on the child, whereas when you control the blood sugar levels, then those impacts, the risk of those impacts disappear, or they certainly reduce. And so what we're finding is that in trials of some of the diseases that are detrimental in pregnancy, even despite the fact that currently we have contraceptive requirements, despite those requirements, people fall pregnant occasionally. Sometimes because they thought they were infertile, because their disease is incompatible with pregnancy and results in early loss. So when this happens, automatically, these women are then excluded from the trial and taken off the treatment. And patients ask us to change that. They really wish to stay on the treatment, accept the uncertainty that comes with that treatment and the impact on the child, and say, this is my choice. And so then my question to the rest of the world is, who are we as regulators to say that they know they need to go back to their standard of care, which was not effective or which is teratogenic. And so I'd like to suggest that we need to find a different balance. So we need to find better ways of balancing the needs for treatment with the uncertainties that come with that treatment about the risk. And so one of the paradigms that we use is we say a healthy child starts with a healthy mother. And the narrative has been that we exclude pregnant individuals from clinical trials because we want to be cautious. But you're not automatically being cautious because the novel treatment may be better than the current standard of care. And well, all the things I've already said. So then my question is, at the moment, what we're doing is we're allowing these products to be used in pregnant and breastfeeding women in a not experimental setting. So the question is it better to experiment in a way in an uncontrolled setting? Because that's what we're doing. So I want to quote Theodore Roosevelt, who says, reach for the stars, but keep your eyes on the ground. So how can we be ambitious, but have scientific and data-driven ambitions going forward? And we have developed at the agency a strategy for strengthening the benefit-risk information throughout the product life cycle. And so I want to start with the post-authorization. So this is specific to pregnancy. So post-authorization is currently what we're focusing on when it comes to evaluating the impact of medicine use in pregnancy and breastfeeding. And so we have a ton of guidelines. We've developed methodological guidance for people who investigate in this area. We have guidelines for industry. And we have tried to develop strengthened signal detection methods. And so what we find is that pregnancy registries, when we ask them of industry, they have difficulty to recruit, they have lost a follow-up, they don't have comparator groups, and as a result, they don't result in fast impact on the labeling. Similarly, post-authorization epidemiology studies, very few make it into labeling. Medics, clinicians, are very good at signal detection, and the signals that we've seen in pregnancy come from the literature. Eurovigilance, our signal detection system, hasn't resulted in signals in pregnancy. We ask companies for periodic safety update reports on their product, how is it used, what are the side effects globally, and so on. We receive very limited data on pregnancy. And the data on breastfeeding is even thinner. So, are we doing the right thing? So, can we change this experimental setting? Is it appropriate to do more in the clinical trial space? Can we generate some more robust evidence in a controlled setting of a clinical trial, carefully monitored, carefully balanced as to when you are more permissive. And that is actually what we've been working with with industry regulators globally over the last two or 3 years. And that has resulted in a draft guideline from ICH, E21, that really, where we've tried to say, okay, with the ambition to be science-driven, responsible, not cavalier, how and when can we be more permissive for to include pregnant and breastfeeding individuals in clinical trials, so that it benefits women who are pregnant or breastfeeding and want to know, can I take this treatment, or how should I, time it in terms of breastfeeding and so on. That is out for public consultation until the 15th of September, and I implore you, we really want to receive your feedback. Let us know what you think. Are we being off our trolley? Have we missed something? What further guidance would you need? And so on. So we really look forward to that. Now I'm going to, oh no, I'm not going to time out yet. What about older medicines? Because basically this is all clinical trials. That's the methods medicines that are coming onto the market now. But most of the medicines used in pregnancy have been on the market for decades. So it's not even one pharmaceutical company that we can ask for the data. Because these products are off patent, it is quite challenging to get data from others. And you also, I wish to really implore you that unless a product is contraindicated in the label, the minute it's authorized for use in adults, it is authorized for use in pregnancy and breastfeeding. Even if the label may say, well, only if the benefits outweigh the risk, talk to your doctor, and so on. So we have an EU framework contract that really enables us to generate data where needed, both in terms of translational sciences along the lines of what we've heard from the previous speaker, and in terms of epidemiology. And you see some logos on the left of the infrastructure generated in conception, built in the COVID studies, and now in studies that really look at the feasibility of when can you and can't you use these epidemiology data for generating robust evidence, because we need to do that with caution. We don't want to create false scares, and we don't want to create a false sense of security. And then I'm going to zoom out to the global picture, away from pregnancy only. And okay, what does this mean for helping better equality in medicines for all women? And so we cannot do this in isolation. Like María said earlier, we cannot sit in our ivory tower, because we only see the dossiers that are submitted to us, and we don't see the products that were not developed because they don't benefit the right proportion of the population, or we haven't identified the benefits for women. So the clinical trial system at the moment gives us structured protocol data, and the studies haven't been completed yet because it was started in 22. Going forward, it will give us the results data, and that will give us the opportunity to really monitor continuously whether the inclusion of women and other groups is appropriate. We have trials map so that people really help enable inclusion for all. There is Darwin and the epidemiology studies that I mentioned earlier that really gives us the infrastructure and opportunity to evaluate utilization and safety aspects of medicines throughout Europe. So we can also keep monitoring and work with our stakeholders where action is required if groups are disadvantaged. So as I said, we cannot do this in isolation, especially when you look at smaller populations such as pregnant or breastfeeding people. To get the data, we need to be global and not Europe only. Medicines development is happening globally. So if we as Europe only have certain demands, that doesn't help. We need to agree with our counterparts in other regions. So we can evaluate the data. We need to work together with our stakeholders to help progress this field. And then hopefully the next anniversary, well, not the next, like the 5 years, but in 30 years, the story will be women and men live for equally long time, and there is, the treatment delay is the same and hopefully negligible because we have AI to do it all for us. And with that, I'd like to thank everybody again for all the work that you're doing on this, that I now get to present. And yeah, thank you very much for your attention.
Many thanks, Corinne. Would you be open to a question? Yeah, there are already questions here. I think, you know, turning 30, then you are at least a grown-up person. And what you showed here is actually, for me, a grown-up reflection and decision-making. Yeah. So I think it shows that the European regulatory system is a grown-up system, maybe even a mature system. So, but anyway, there are some questions here for you.
Thank you. So considering, I won't read it out, I mean. Well, what have we missed as regulators? I'd like to think that we are now aware of this and we're not missing anything as regulators because we're working on it, right? So we have got the framework contracts in place, we have got the Darwin system in place, we have got CETIS and the clinical trials map in place, so that going forward, we will do better. And if in the feasibility studies, we can demonstrate that with the infrastructure in place, we can do comparative studies of, for example, anticonvulsants or antidepressants in pregnancy, where at the moment, if you are pregnant and you have major depressive disorder, there is no medicine for which we know this is safe. So you have to choose to take a medicine with other therapy, or not have the medicine, with the risk, of course, that if you have major depressive disorder, that you have a suicide attempt, which in women is usually with an overdose of medicines. So, which one do you then take? We don't know. And if we can demonstrate that we have the systems in place to do comparative studies, then my plea to the powers that be at this agency, and I have a listening ear, so I'm hopeful that this continues, is that we should be doing that kind of work. So we should, if it is feasible, because we don't want to be creating false alarms, if it's feasible, we should do the comparative effectiveness and safety studies to help patients. Because from my time in academia, I know that charities won't fund it, because they fund the work to support the affected children and the affected mothers, and they say this is for industry to do. Industry doesn't do it because they have a mandate for their own product and they don't wish to compare their products with the other ones, especially in this sensitive space. So this is for me an opportunity where we as regulators can step up in a responsible fashion. If it's not possible with the existing systems, then we need to think again.
Many thanks, Corinne. I think we will leave it by this, and thank you once again for presenting. So the last presenter is Marco Cavalieri. Marco is, I think, known of most of you, if not all of you, head of public health threats here at the agency. And you and I, Marco, we belong to the group of people who talks about when the next pandemic comes and not if it comes. So therefore we ask you to talk about how are we tackling the next pandemic, and yes we can, so we can end on a positive note here. Marco, the floor is yours.
Thank you very much. It's my pleasure to be here. And indeed, you know, the motto here of this 30th anniversary is "the future is now," which is great. But here, you know, do we really want the future to be now? Because, believe me, another pandemic will happen. Epidemics will happen. So we need to be prepared for that. And we need really to work in the direction of doing the best we can to tackle the emergencies of the future. Why am I saying that? Well, first of all, epidemics and pandemics have always occurred since human beings have been living on this planet. That's what we know from the past. But what we know that we are making changes to our planet that will impact on the frequency of these emergencies and these epidemics. First of all, as you see clearly here on the left, the world is becoming quite crowded. You know, the population has been really increasing massively in the last 100 years and is going really steeping up in the future. So we need to think about that, because the more people are around, the more chances are that transmissible diseases will be expanding and causing emergencies or new waves of infection. So that is a fact of life that we have to cope with. The other point is that, of course, what we are doing to the planet, you know, deforestation, destruction of the wildlife haven, all this will lead to more chances that a number of these pathogens that are living placidly within the wildlife could come to us and could cause new problems. And then, of course, global warming. And, you know, it's not about, you know, temperature is growing 1 degree, half degree, and all the debate that is going on in the world. Look at the mosquitoes. The Aedes albopictus mosquitoes, which are the one that carries viruses such as dengue or chikungunya, now they have come as north in Europe as Brussels and Berlin, which means that in a few years we'll be here, maybe in this room. We will have the Aedes albopictus around us. And with them, the virus will come along. So we need to think about the fact that the future, in this sense, might be more problematic or more complicated. So we really need to think about the fact that in the future, we will have more chances of seeing epidemics than what we've seen so far. Another point that, you know, I could not resist talking about is SARS-CoV-2 and the origin of the virus. Now, as you know, there are some people in the world that think that, you know, if you shut down laboratories that are doing research on viruses and you get rid of this called gain of function type of work, then we will not have a problem with pandemics in the future. Well, we should not fool ourselves. The reality, as the papers that are, you know, on this slide are showing, is that the ancestor of SARS-CoV-1 and SARS-CoV-2 have been found. And these viruses, they live in China and Laos. And they live there since many, many years. Actually, the virus that they gave rise to SARS-CoV-2 was around in these regions between 1 and 5 years ago. What was really astonishing is that, you know, these viruses, they are commensal to the horseshoe bats, which are a certain type of bats. And again, they live in their gut and the oropharyngeal tract of these bats without causing any problem. You know, there is no issue there. The problem is when the bats get in contact with intermediate species, and there is a lot of wildlife still there, and then, you know, these intermediate species are the one that then led to the transmission, or most likely is the real reason of the pandemic with COVID-19. So essentially, the virus was in the bats in that part of China, and then all of a sudden, because of intermediate host that was pretty much identified for SARS-CoV-1, but not yet for SARS-CoV-2, ended up with a virus that also replicated in the mammals, so really adapting to mammals, and then gave rise to a virus that was the one that caused the pandemic. So we really need to put efforts in continuing the work in laboratories, not stopping this important work on gain of function, but also complement this with work on surveillance. So the so-called One Health concept that Sandra mentioned before is extremely important. We really need to continue surveillance, understanding what is happening out in the wildlife, and really spot and monitoring what is happening with all these viruses. And I will just quote SARS-CoV-2 also because what these researchers discovered, that there are hundreds of sarbecoviruses, so coronaviruses similar to SARS-CoV-2, that are living in the bats in this region. All of them could cause a pandemic. And there are thousands of coronaviruses in bats all over the world that could cause a pandemic in the future. And this is just one example. There are many others. Avian flu, you heard a lot about already today, so I will not enter into that. But there are many pathogens that could cause problems in the future, and we need to be prepared on how to deal with all of them. So now is the problem statement, and, you know, and why we need to take action. So what are we doing here at the EMA, and what we have been doing? So here, the EMA, as you heard, there has been an emergency task force that was already creeping up over the years. It started working actually in 2009 with the influenza pandemic at that time. It was very embryonic, but was a new way of working within the European regulatory network. So about disruptive regulation is also how you organize ourselves and how we do the work in Europe is very important to look into new approaches and new way of working. And the emergency task force, as you know, started working at the beginning of a pandemic, even without a legal mandate, simply based on an health threat plan that, by the way, the EMA had, was one of the few institutions in the world that had an health threat plan already established just before the pandemic, so probably we were lucky, but it was there. And so we could activate that and start working straight away through the emergency task force and conducting a lot of activities on how to handle the pandemic. But I'm not here to talk about the lesson learned of what we've done. I'm here to say that the emergency task force now is established and it has an important role in looking into whatever is popping up, whatever it will be coming, whatever the future might bring us in terms of epidemics and pandemics, and be as prepared as we can. So doing all the work that is needed in order to have Europe and the world better prepared to face future emergencies. And it's not just pandemic and epidemics, which, you know, it's Mother Nature's that will bring to us, sometimes instigated by human beings and by what we do to the planet Earth. But it's also about the direct effect of humans. And that's why we're dealing with the so-called CBRN spectrum, which is chemical, biological, and radionuclear, which is very important because, you know, the world is very unsettled. There is a lot of wars going around, and we don't know what the future will bring. So we need to think about also the possibility that some of this threat will come deliberately from release from human beings or from an accident, like, you know, a nuclear disaster. So we need to think about that, and that's what the ETF is doing, to really think how we can be better prepared in having the right medical countermeasure to face all this issue. And then there is this big topic of antimicrobial resistance that is so big that I will not even dare to enter into that because it will dedicate, you know, an entire symposium. But you know, and you heard today how important it is. María, Sandra, among others, mentioned that. So we need also to work on that spectrum of what is called the silent pandemic, which is not silent at all because people are dying when there are no antibiotics available that work. So we need to do something on that. And even if it's moving at a slow pace, it's not like COVID-19, something abrupt that, you know, involves millions of people straight away, but still is there and is a real problem. So we need to do something about that. And the emergency task force really works in three key areas. The first one is to provide scientific advice and support to developers, to academia, to whoever has a good idea of advancing a medical countermeasure that could help us in dealing with these emergencies. And you see here some figures which, you know, when I read them, I still don't believe that, you know, we had like 250 plenary meetings during COVID-19, and we processed, well, some of them was a bit of a fast process, I would say, but, you know, 270 therapeutics and 80 vaccines. These, you know, are incredible figures and really shows that in a pandemic such as COVID-19, the amount of work that will come to the EMA is impressive. And we need to find a good way to process all of these and to work through to make sure that the best candidates can advance as rapidly as possible. And now we are continuing this work on scientific advice because preparedness is key, as I will say at the end, and we need to make sure that medical countermeasure, vaccines, monoclonal antibodies, antivirals get advanced as much as possible so we have them on the shelf to be prepared for the next emergency. And then scientific reviews. We did a lot of scientific reviews during COVID-19. You remember every day there were hundreds of papers coming out as you know, preprint, and we had to look at them also because sometimes journalists ask us about papers that we are not read yet. So, you know, was really a race in getting out with information as rapidly as possible. But you remember hydroxychloroquine? Well, that's something that I'm very proud of because the EMA has been the first agency in the world to come out with a clear statement. It was May 2020 when, you remember, on the other side of the Atlantic an emergency use authorization was granted to say don't use hydroxychloroquine except in a randomized clinical trial. And we were the one to go out with this loud and clear message. And that's what the ETF should be doing. That's what we should be doing as European network. Come up with clear recommendation to patients and people all over Europe and all over the world. And I have to say, you know, Thomas, that if we had some organoids, maybe with a sprinkle of artificial intelligence, probably we would have understood that hydroxychloroquine was not even a good agent to start with and should not have been even prioritized for clinical trial. But that is another topic, and we will have to deal with that. But of course, we need to take into account the innovation that comes from this type of approaches and use it more in the future, because that's the way to go. And then giving scientific recommendation, we made a lot of recommendations on the use of the vaccine, jointly also with the European CDC. And also recently, for example, the ETF came out with a recommendation on how to adapt the COVID-19 vaccine, which COVID is still around, you know, it's not a threat as it was in 2020, but still, you know, for elderly people and immunocompromised, it's still a threat, so we need to protect them, and we need to think how to protect them in the best way possible. And that's what we are doing through the ETF. So come out with this recommendation, which, again, is outside the ordinary of what the EMA used to do. It's something new, but so badly needed. European citizens want to hear a strong voice from European institutions that is credible, that is science-based, and help them in understanding which medicine to use and how. And then, you know, the pandemic is over. We had the fantastic, you know, achievement of having vaccines approved in 10 months. So, you know, we've never seen such a fast development. And again, no corners were cut. All the clinical data that we need were there. Simply everything moved at a pace that was before we had never seen it. And that was great, but that was 10 months, and it looked like something very difficult to beat. Now, the idea that has come up, and this is a document also coming from the G7, G20, is the goal of getting vaccines, diagnostics, and antivirals approved in 100 days from the start of an epidemic or a pandemic. Well, this makes us regularly a bit anxious, because, you know, to get there is not easy. But this is aspirational, and I like the challenges, so we need to work on that, and we need to think how we can get as close as possible to achieve these 100-day missions. How this is going to be done? So let's think about the so-called pathogen X, which is, you know, the unknown, and a new pathogens that will cause a pandemic. SARS-CoV-2 was a bit of a pathogen X, because it was not really on the radar of most of public health authorities. And therefore, you know, how we can get as close as possible to the 100-day mission. So the concept is to advance prototype vaccines and monoclonal antibodies for defined priority viral families. So in other words, the WHO, with a group of experts from all over the world, and we participated to that, defined 12 viral families that are the one that most likely will give us a virus that will cause an epidemic or a pandemic. And then within the families, single viruses were selected as prototype viruses and used them to build vaccines and monoclonals. And this is taken into account with all the new technologies that we have. And you see here on the slide some example, you know, using a single lymphocyte B to select a new monoclonal antibody, use the new way of constructing antigens. For example, do you know that for SARS-CoV-2, essentially, most of the developers use a way of designing the surface antigen of this virus using the experience with the respiratory syncytial virus. So stabilizing the conformation in a so-called pre-fusion conformation, which gives better immunity. All this is very important, because taking together all this scientific advancement, we can be faster in coming out with this prototype vaccine in peacetime, and maybe get to the point that they can be approved, and at the ETF we are ready to support this and to see how far we can go. Ideally, and I think this is really the ambition, is that for some of them we can come even to an approval. And this, I have to say, we'll have to rely on animal models of infection, because it's very difficult to do otherwise today. I don't know, Thomas, let's see in the future, but for now we are really stuck with that. But the idea is really to advance as much as possible so that the prototype vaccines and monoclonal gets approved, and then when the pathogen X arrives, which will be different but belonging to the same viral family, it will be closely related. So with some few adjustments, we will end up with having the vaccine and the monoclonals that we want. And that will take less time. That will take less clinical trials and less evidence. So that is the entire strategy that we are working on in an international landscape, of course, and that's what we are aiming to do in order to be better prepared for the future pandemics and really try to achieve this 100-day mission. And of course, international collaboration. You know, the adage goes that no one is safe until everybody's safe. So that's true. And we at the EMA, we always work internationally. And during the pandemic, we had fantastic example of international collaboration. I'm quoting here with the ICMRA, which is a great platform for international discussion. Agencies from all the continents in the world are represented there. And it was fascinating that during the pandemic, we felt it was so difficult to have an agreement globally with all the regulators in the world on the study design for new vaccines for COVID-19. And you remember this also the media were picking up on this and tried to look into differences. And, you know, at the end of the day, everybody agreed. So we came out from that workshop with the entire world agreeing on the study design for vaccines and for therapeutic in another workshop for COVID-19. And that was great. That's what we need to achieve during a pandemic. We need to achieve global alignment to the extent possible. And then, you know, another example is this one, because now there is, after the WHO defined this viral family, there has been the creation of this collaborative open resource consortia, one for each viral family. And for example, here I'm putting up the one on flaviviruses, so you know, dengue, Zika, and all these nasty viruses that are mosquito driven. And Fiocruz in Brazil is the lead of this cork, and EMA is in the lead of the regulatory activities for this cork, which is extremely important. And we are involved in many other corks. So really here is international collaboration with the regulators, but also with the research community at length. So we need to work with all the world in all different areas of research and development. And that's what we're doing, and that's what we need to continue doing for the sake of a good response in the future. And then let's not forget about, you know, re-emergent infectious diseases. Mpox, that is a very good example. Mpox re-emerged because simply the vaccination campaigns for smallpox, which led to the eradication of smallpox, big success of vaccination, you know, eradicating a disease such as smallpox, believe me, it's a really great achievement. But then, you know, once smallpox was eradicated, of course the vaccination campaign stopped because there was no need to vaccinate people for smallpox. And then what happened that in the DRC, when Mpox is around, this came up again, particularly in younger people, because younger people were not vaccinated for smallpox, they get cross-protection also for monkeypox, and therefore, you know, Mpox re-emerged. So we need to also keep in mind that if we don't want viruses to re-emerge, we need to maintain our capacity of keeping them down or avoiding that, you know, they will come back. And a very good example is polio. Polio, we have seen polio in wastewater surveillance, so you heard today about the relevance, you know, after COVID-19 of doing this wastewater surveillance, and polio has been spotted, you know, in Spain. Many cities in Germany had polio spotted in wastewater, and polio dries very fast, is extremely transmissible. So can you think, if we do not maintain our vaccination coverage of our children, what is going to happen? Polio will come back. And it will be an unacceptable tragedy if we see even a single child dying of polio, which is a completely preventable infectious disease by vaccination. So we need to maintain our efforts on the value of vaccinations, the importance that children, elderly, adults continue to be protected with the vaccines that we have, and we should not lower the guard at all. And we should resist of any miscommunication or misleading message on science here. So very important that we don't forget sight of this. Then moving to another topic very rapidly, which is very important for us at the EMA, clinical trials. During the pandemic, there were so many clinical trials conducted that were of poor quality, unable to answer any question, completely useless for patients, researchers, and authorities. And all these studies were approved, get rapid approval, while the big studies that were multi-country, that were able to answer many questions, had a lot of issues in getting approval. So this is something that we need to change radically. We need to do better, particularly in an emergency, to have timely clinical trials run, conducted, and delivered so that we know what to say to citizens and to patients about what to use and what kind of drug to use. And here at the EMA, we are taking a lot of activities. You know, it all started with an important workshop with the scientific community and with many public health authorities about what happened during COVID-19, but also during Mpox, because during the wave of Mpox in 2022, we see also a big difficulty in running large clinical trials in Europe. So we need to do better, and you heard from María, ACT-EU as a priority action on clinical trials in public health emergencies, and we are working hard here to make sure that we have a more flexible approach to the approval of clinical trials, a faster process, a streamlined process, and also this concept of a one-stop shop in which developers, investigators, academia come to us through the ETF and get a feedback that includes not only the view of the ETF, but also the national competent authorities that have to approve the clinical trials, and also from ethics advisory group, because we also want to embed there the view of the ethics committee. You know, we don't want to end up with something that maybe is okay for us and for the clinical trial unit in the member state, but then the ethics committee a problem, because that will stop the process. So we need to do better, and this concept of centralizing at least the advice in order to make sure that there is clarity by all parties about what is needed is so important. And then, you know, the fragmentation in terms of conducting clinical trials in Europe has been a big issue. We need to do better. We need to work with European institutions, with DG ERA, with RTD, with Santé, in order to make sure that we have a good framework for coordinating clinical research and prioritizing really the clinical trials that will give the answer that we need as rapidly as possible. So we are really working hard on this, and I'm quite optimistic that we will see much progress in this area and Europe could do better. And then another important point, you know, clinical trials are essential, but also it's important about other type of evidence that needs to be collected during an emergency, such as a pandemic. And you remember very well during COVID-19 when we were thinking about when to give booster doses for the vaccines, when is the right time, should we change the composition? Well, we all relied on these effectiveness data that were coming from our other regions outside of Europe, you know, Israel, UK, US. How can it be that in Europe we are not able to conduct this type of studies? We should be able to do that. We need to be able to do that. And therefore, you know, together with the European CDC in a joint venture, we established this vaccine monitoring platform. There is really the ambition of creating a large network across the European Union so that we can conduct very rapidly this type of effectiveness studies or on safety of vaccine, so that we can understand how to use the vaccine, how to inform the public, how to inform the people in Europe about how to use this type of medicinal product. And here, this example is quite emblematic because it's after COVID, you know, it was Mpox. And in 2022, we approved this vaccine, Imvanex, for Mpox, but there were no really efficacy data then. And we wanted to have some data coming in terms of vaccine effectiveness, but there were no funds available in the European system to fund these studies. So we were blocked. We could not do much. So the EMA bravely took the opportunity and the burden to fund this study. And this study delivered data that is extremely important. You know, Charité in Berlin was the principal investigator of the study. And this study really told us what we expected, that the vaccine is effective, and even a single dose, because it's a two-dose regimen, even a single dose provides more than 60% protection. And this is really important. So we need to establish this in Europe. We need to establish our capacity of conducting these studies during emergencies and also in peacetime, I would say. So the key message is that we got now the ETF, which is legally established with a new mandate, and these are new tools to facilitate and accelerate the approval of medicines during emergencies and building on the experience on COVID and Mpox. Preparedness is really key, and that's why we're really working on this coordination of activities, which is in Europe. It's not just industry, it's also academia, and it's international, and we need to work on this in a very flexible way to get meaningful results. Really facilitating large multinational studies is the direction that we should be taking. We need to have the infrastructure, the networks, and the capacity of the regulatory networks of rapidly approving these studies whenever an emergency comes up. That is the only way to generate robust evidence as rapidly as possible and improve coordination and collaboration. As I said, with academia, we're working a lot, but also with non-governmental organizations, SEPI, Gates Foundation, and others. EU bodies, of course, national competent authorities. I think during COVID, we had fantastic interaction with the member states and at different level, public health authority, regulators. That needs to be continued and it needs even to be improved. So there is a lot to be done, but we know what to do. And that's what we're going to do. So the mantra was, is, and will be that an effective response comes only with effective preparedness. If we will be able to put resources, funds, efforts there, we will make it. And we will, what we did already with COVID, we will do again and again and even better. Thank you very much.
Thank you, Marco. I think you answered the question, yes, we can. I feel confident. I think we have a single question, or maybe more, but I think we have time for a single question here about the WHO pandemic treaty. Is it part of getting more tools in the toolbox?
I think it does. You know, I think it's very important that we have a treaty. And for me, the bottom line is not the treaty itself, but the fact that countries in the world understand the importance of working together. And working together has to be built on trust. So if we trust each other, we will be able to do it. And that's the principle in the pandemic treaty. So that's why I see this very positively, because it's an open door to really foster this climate of collaboration and trust. Without that, I don't think we will go very far in terms of pandemic preparedness.
Completely agree. Thank you, Marco.
Thank you.
Thank you. So this takes me to the end of my task as chairman of today. It's been a pleasure. Thank all the speakers. I've got to thank all colleagues who have helped me prepare the program for today over the past, I don't know, a year or so. Colleagues who have spent many days in preparing for all the practicalities, making sure that His Royal Highness comes in at the right door, enters the right steps, coffee tables, coffee, lunch. EMA for giving me the challenge and the task to create this day. So I'll leave the floor to Rui, who is president of the Portuguese authorities, but you are here also as the chairman of our management board to say some closing remarks. And I know you prepared a couple of hours speech here. And when those hours have gone, there will be a networking reception afterwards. Thank you.
Thank you. Thank you. Yeah, thank you very much, Stephen. I think you've given me the most difficult, simple task, because it seems very simple just to say a few words at the end of the session. But as you said, we were planning 2 hours speech, but then we prepared something in between. But now I feel that I probably will try to even do something even in between the between. So I will use my notes, but I will try not to go through them, because I think we lived a very important day, very emotional on one side, very rich on the other side. So I think, dear colleagues from the EMA, dear colleagues from the network, dear guests, dear all, I think we should be very grateful for the day that we have had today. It was really, really, really important, and so I will come to the thanks at the end for those who were behind this organization, but I have to acknowledge this at the very beginning, my deep gratitude to every one of you, not the ones that were organizing, the ones that have been here. The first reference I will make is what we had this morning, still in the presence of the King of the Netherlands, or His Majesty the King of the Netherlands, with this inspiring lesson, and Professor Yann Holst is still here, where he told us on how the basic science is essential, in this case, to make new therapies a reality for patients in the area of diabetes with GLP-1 receptor agonists. But he also showed this, and also what are the potential areas that are ahead, also making this very interesting connection with this playing a role as incentive to a better lifestyle. So I took good note, Stephen, as you, to also improve that aspect, because I think I need a little bit. So this was the first remark I wanted to make because I think it was very, very important to start this scientific conference, EMER, this morning. Then I would like to perhaps make some references to the past, because you gathered this today in this anniversary in this remarkable journey. I think all those that have been involved are here. So we have all the former executive directors who were participating. Unfortunately, Fernand Sauer was not able to join us in person, but he was there. We have Thomas Lundgren is sitting in the room. Where is... Thomas is there, yes. We have Guido Rasi, we have Fernand Sauer, we have Andreas Pott also there. So all the executive directors here, and of course, Emer, you are the one that came when there was already this challenge, because you came through during the pandemics. But I think also this meeting today shown that challenges are not ending, so they are probably increasing more and more. I mean, María made also this very visible. So my special recognition to all of you, because you have been leading the agency through these different phases, through different pivotal phases. So you were responsible for where we are today. But I also would like to acknowledge some other colleagues. Forgive me because I will not mention everybody. I'm always trying to do that. Also, my colleagues know that I do that at home, and I always fail because I always forget. I think Emer and María have witnessed that. But I think we should, because also there are many heads of agencies, not only the ones that are today in charge, but also many from that are former heads of the agencies, national agencies, members of the management board. I mean, many are the same. But I would like to refer to Sean Heppel, that is here, the first chair of the management board, and also Krista Virtumer-Hogg. She was not only the chair of the management board quite recently, just before Lorraine, and Lorraine is also here. But Krista has been also part of the foundations of the system, because I should not say that, but you have been during a few years around the system. Forgive me also to mention my colleague, but my friend, Aranda da Silva, José Aranda da Silva. You heard Fernand Sauer mentioning, because they are very good friends. But Aranda was also very much involved in the setting up of the agency. He always tells me, I think this is true, that the political agreement was achieved during the first Portuguese presidency in 1992, and the regulation came in 1993, as you know. But Aranda was behind that. Aranda and Agnès Artige, that was also, it's also a bit emotional for me, because I have a lot of connection with the EMA in different phases of my life, also because probably not everyone knows, but many know that I have also been working for the agency between 2000 and 2002, still in the West Ferry Circus in Canary Wharf. So this were the, I mean, of course, there are the colleagues that the team of the EMA that were at the very beginning. I think some are here, some came, and some probably are still here, because I think there are still a few from the very beginning. So also my recognition to, and this is also to connect with one point that Guido made in his presentation, that these 3 decades have shown that these people that committed, visionary, resilient, because you were probably the one that came through a good part of that resilience. I mean, Emer took over in between, because 2020. But you said that these people that make the difference, and I think that is not, we should be reminded, all of us, that is not the structures, but the people who shape the path forward. So, and this, I think, the history of the EMA has proven it. So I would like also to mention that aspect. Of course, there are many other people I should mention. Sandra Gallina has just left. I will refer to her in another point. And then also, of course, a very special reference also to María. María, I interrupt you now, María, because you are here in different heads as well. You are the head of the Spanish agency, but also the chair of the management group, and also a very inspiring colleague that is always challenging us, and you also did it today. So, María, thank you very much for having this very important way of intervening in the context of this network. You said that you would not be able to predict the future, but you would be able to at least to influence or to shape it. So I also can agree with that. We also heard that through the voice of Sandra Gallina, that Commissioner Várhelyi, I cannot pronounce the name, Várhelyi. I know it's not like that, but was also putting a lot of emphasis on the important role of the EMA in the European Union, and also in relation to the new legislation. So I think this is also very important, that we are building a future-proof system, a future-proof system that is based on science. And science, this was very much mentioned during the day, that with Emer, you started with that, but also María, and now during this last session that we just heard, we were making all the bridges on all the aspects about, so that science should be a fundamental base, Guido mentioned that as well, of the work of the agency in relation to what are the answers that we have to give in terms of innovation, in terms of better regulation, in terms of competitiveness, and all this. I also had here, I'm jumping a note on from Yann Lecam, because you, Yann, also emphasized and reminded us importantly of the importance of the engagement with patients, and I think this is also very important that I underline that aspect, because also I think the EMA was pioneer also in this work, and it continued evolving up to now. We have to find a good solution in the legislation, as you said, but I would like to mention that aspect. And here I also would like to come to a reference to the scientific committees, because the scientific committees are also an essential pillar of the activities of the agency and the network, because we all together rely on the scientific action, scientific base that comes out of the different structures that are here at EMA. I know that there are, I had a note here. Yeah, I think there are, all the chairs are here, I think. So the cat, Ilona, Brian, PDCO, so the Compis team, also Violette and Bruno are here. They were former chairs. We have also the chair of HMPC, Emil. And now I cannot read the chair of the CVMP, Grit. Johan. Sorry, then I apologize. And Prag Ule is here, I know, and also I think Sabine, and also June Rain. And allow me also to say a special word to June Rain, that is, and all the colleagues, all the current chairs and the former chair. Of course, Bruno, the current chair of the CHMP. I was forgetting him, probably because he's Portuguese, but should not do that, because he will have more work in at least in the coming years. Not sure exactly when. Then I wanted to make a referencing. This was very much shown in this last session, and Sandra Gallina also emphasized, and I think Emer as well. It's the importance of having this more holistic view and a more global vision in terms of public health. Not only, and also the sessions in the afternoon were also bringing us into the different aspects of the science and how they can converge into our responsibilities, but also the role of the agency, the EMA, in the international context, and of course this links a bit as well with the disruptors that you mentioned. But I think this is very important to underline also that EMA evolved to be a very respected and global player that will need to continue. And of course, when it comes to issues like preparedness for pandemics and things like that is very important. A final words, I think, on looking a bit more, looking to the future. I took here another note from Guido, because you said fighting inertia, and I think we all agree that inertia is something that we have to combat. And when we are thinking to look into the future, to keep the vision, to keep the commitment, to keep the system responsive together with the network, because I think this is also a distinctive element of the agency. And this, again, if I can go to history, this was also a strong discussion in the 90s, or before probably, because the agency, as you probably remember, it was part of this great vision of the internal market, also in 1992. So, but it became as a networking agency, and I think we also were able to develop it as such. And if we look into the documents, we see that very well reflected. And I think this is a very important element that we have to preserve and to use on the benefit of the public interest. I had here the reference to many of the things that are ahead, and many pieces of legislation, some that are more specifically direct to us, but others that are more global, but they are also very important. But I would not mention them in detail, but of course we have to be more and more conscious that the activities of the agency and our role is going beyond what is, let's say, what would be the typical role of the regulator. And this is very well reflected as well on the strategy that María referred to in her intervention, when going through these 6 topics, we see that our role is being closer to the ones we serve, to the public interest. I'm now closing, but I would like to close with 2 references. One is again is really to acknowledge the dedicated teams that are behind this event. I would like to express great thanks to all of those. Perhaps I name 3 names: Emer Cook, Ivo Klaassen, and Stefan Tirsup, but of course I know that behind I was just sitting with Melanie on my line, but so thank all of you, because not only those that were in the program organized, but all of those that were behind. I mean, Stephen was alluding to that. I wanted to associate myself very much to this, showing this capacity and professionalism that we could witness today. I could not also being here in this closing session of the 30th anniversary. Fernand Sauer made this allusion to pay tribute to important persons, important friends of us that have played an important role and that unfortunately left us very soon, very unexpectedly, both of them. So Joseph Torrent Farnell, the first chair of the COMP, and Noël Watillon, that is now in the name of this auditorium. But I've known Noël for many years. I think all of you have known Noël for many years. We have also, of course, worked together. But I think his deep sense of service and commitment to the agency's mission, because he kept staying through Brexit and he kept staying through the pandemics, is really enshrined in the evolution of the agency. So I would like to publicly recognize his legacy on this occasion. And now, just not to repeat what I said, when we look to the future, perhaps now I read a little bit because it probably makes sense. Let us carry forward not only the lessons of the past 30 years, but the enduring values that have brought us here. Collaboration, integrity, scientific excellence, and the unwavering commitment to public and animal health. Also, it was mentioned the geopolitical situation we are in should also position the EMA into a new dimension by making a strong call also to reinvent itself, to look into things that are being looked, I know, to the procedures, continue embracing technology across all responsibilities, and this was also very clearly shown during the session. As I also said, the European Medicines is not just a regulatory body, it's a scientific community, is a guardian of trust, and this should be well kept. I conclude by thanking you all for your contributions, for your service, and for being part of this extraordinary journey. The next chapter begins also with us. Allow me just to say, because we have here the logo of the agency, Science, Medicine, Health, and I noticed, Emer, that on the program there is medicines, regulation, and future. So I think the two combined really can be a very good conclusion for today's, because I think this was very much connected with all these 5 words, I think, because yes, 4 words. Thank you very much for giving me this opportunity.